Welcome everybody here in the room and everybody virtually joining us online. Delighted to welcome you all to our annual Benioff Center for Microbiome Medicine Symposium. And this year, the topic is Interdisciplinary Approaches in Microbiome Research. My name is Sue Lynch. I have the pleasure of serving as the director of the Microbiome Center. And over the past 5 years since we launched, we've just been growing from strength to strength. The vision of the center originally, as we considered it, was to really build up this field at UCSF and to democratize the microbiome research field across campus. And so to do that, we've built quite a lot of research infrastructure. We've got a brand new notobiotic mouse facility that is commensurate with the level of exceptional work that goes on there, led by Jesse Turnbaugh. We've built out our microbial genomics facility, and we just recently received an award to build out the quantitative metabolomics analysis center, and that will be built out and open somewhere around 2028. In addition to that, some of you might have noticed the very large hole on the west end of campus as you came in. I've never been so happy to see a large hole in the west end of campus, but that is the site of our brand new research and administrative building that is going up and will also open in 2028. And a large portion of the Microbiome Center will be relocated and co-located in that building with many of our other sister programs on campus, diabetes, ImmunoX, and several others. So we're pretty excited for where microbiome research has come from and where it's going over the next 5 years on campus. If the first 5 years we're building up the community of now over 200 members on campus, the next 5 years are building out and really developing larger research initiatives with partners across the country to leverage what we've built here and to really attack head on the key issues that are to be addressed in human healthcare. And so a couple of things before we get started. First of all, I want to thank the speakers. I mean, just an amazing lineup that we have today. I've never heard so much excitement about one of our symposia. For those of you who are local, you know, we have Outside Lands, and it's just amazing lineup. I feel like this is the Outside Lands of microbiome research. Just incredible to have all of these wonderful speakers on site. It's just amazing for us. We're very grateful for your time. We know you're all very busy and very grateful for your time. Also, for those of you online, thank you for joining us. We couldn't fit everybody in this room today because of all of the construction that's going on campus. We're kind of limited in the rooms that we have available to us, but we're glad that you can join us even if only virtually. A few housekeeping items. We're only going to take questions in the room because there's so many people online. We know that we wouldn't be able to get to everything. So if you do have a question in the room, please raise your hand. We'll bring you a mic so that those online are able to actually hear the question. And then for the speakers, the one thing I will ask is we've got a full slate, so please try to stay on time. Slovena will be your timekeeper sitting here in the front, and she'll give you the notifications of how much time you have left. And then I obviously to pull something like this together takes a Herculean effort. I want to especially thank Kristen Dolan, who's up at the back, our assistant director of the Microbiome Center. And Slovena, who's here in the front, our communications officer for the center. And the community. We love democracy in the BCMM, and the topic for this year was really chosen by our community, and the speakers were chosen and suggested by our community, and thank you for your interactions and your suggestions. We hear you. And lastly, I also want to thank Dorothy Wong, who's helping keep everything move smoothly on the AV side. Thank you, Dorothy, for your efforts as well. And so we'll have a number of our executive committee and Margot Bastino, who is our trainee ambassador, to introduce the speakers in the session. Margot is what we call a trainee ambassador. That means that she has the great job of figuring out what all of our trainees need and then bringing that to the leadership of the center to solicit funding so we can build out programs to enrich our trainee community within the center. And so Margot, the floor is yours.
There we go. Excellent. Thank you so much. Let me see if I can get my screen share. And while this is coming up, in as much as I have a love-hate relationship with our institution, sometimes I should give Harvard credits, the Harvard School of Public Health, rather than Stanford. But I also changed my title a little bit so that I can talk about a few new programs that have been getting off the ground, all of which will have some interesting acronyms that we'll run across today. So one that's really just started in the past few weeks, I'm delighted to give one of the first talks where I got to mention the Human Virome program. So in as much as the NIH is having a rough month, a week or two ago, we were able to announce the launch of the Common Fund's Human Virome program, which has been planned for several years now as a way to explore one of the sort of underappreciated or understudied areas of both human-associated microbial communities and microbial communities at large. I am delighted to be working on the coordinating center for the HVP along with Owen White at the University of Maryland. So we'll get to do some fun things like managing the data and tool availability from the project, some less fun things like running the meetings and the logistics. But it allows us to work with a consortium that consists of a few different types of projects that the NIH has organized for the HVP. There are several virome characterization centers, or VCCs, based at 5 different institutions, more plus different institutions across the country. These focus on acquiring samples and virome-targeted data from large populations. There are a smaller number of tool and characterization or functional studies as well. These target specific bioinformatic or wet lab or biological areas of interest around viral community characterization. So you can think of this as a pipeline in which the characterization centers have the mandate to make new data that will shed light on the human virome. The tools and protocols will help with new methods for doing that better. The functional studies are meant to deep dive into particular biology center will be to really help get these resources out to the community. One of the areas that, of course, my group focuses on in general and that we'll be helping with for the HVP is in bioinformatic methods. And for a while now, we've been working on improving methods for virome profiling with or without additional wet lab protocols like virus-like particle enrichment. Of course, viral isolation by itself from communities is extremely challenging and in many ways even more challenging than culturing difficult microbes. And VLP protocols vary a lot in their quality. Some can be very good and some can not enrich viruses quite as much as you might like them to. So to address this, many groups for the past few years have been looking at ways to get more virome data out of normal metagenomes and metatranscriptomes, which have resulted in some really great virally targeted assembly catalogs or VMAGs, metagenome assembled genomes. This works really well for virus discovery. So you can fish out new viruses this way, but we know we'll never see all of a microbial community, be it viruses or bacteria, using these kind of assembly targeted methods. Community assembly works great, but for less abundant community members, you simply can't have enough sequences to assemble everything out. So this led us a year or two ago now, in preparation for the HVP, to working on ways to get better viral profiling out of typical metagenomes and metatranscriptomes. So we've just finished up a tool called Baklava that, like all of our tools, has a complicated acronym of its own. The goal of which is to get quantitation of viral taxa from typical short-read shotgun metagenomes, or of course RNA viruses from metatranscriptomes. To give a quick overview of the methodology, Baklava combines 3 modules, one of which leverages nucleotide-based profiling that shares a lot of algorithmic similarity with our Metaflan tool for microbial community profiling. There's a second layer in Baklava that leverages some concepts from our Human tool for functional profiling, in as much as Human tries to find protein families from metagenomes and metatranscriptomes to do things like metabolic reconstruction, we can use that same broader protein profiling to find viral proteins that might have less direct homology than we need for nucleotide matching. And there's a third arm in baklava that can leverage assemblies to do basically virome contig classification, very much like what tools like VirSeq and some of the deep learning tools for viral classification currently do. What this provides out the bottom then is an integrated virome taxonomic profile given a shotgun metagenome or metatranscriptome. And I should call out before I go on too, this has really been the brainchild of a PhD student, Jordan Jensen, whose face you'll see on some of these slides. So in order to enable this, we've put together sort of the largest integrated viral genome bin database, or VGB database, that we can by integrating several of the recently published individual viral MAG assembly databases. Some of the genomes that we have as input come from really gold standard sources like NCBI or ICTV. Others come from several public VMAG databases or our own virally targeted assemblies with Nicholas Agata's group, leveraging some of the workflows he's put together for clustering together similar MAGs into species level genome bins for typically bacteria, or in our case, viral genome bins for viruses that are kind of sort of at the species or genus level for viruses. So to very quickly summarize this, we input about a half dozen different VMAG and viral isolate genome databases. We cluster those into viral genome bins that represent roughly individual viral taxa. There's a super clustering step that handles things like genomic rearrangements that are much more common in viruses than in bacteria. And then we can propagate taxonomy from the more reliable sources to the less reliable sources, and use a combination of nucleotide marker discovery and protein marker discovery. This allows us to capture both kind of better characterized viruses and more genomically novel or rearranging viruses. And as a result, and I apologize if these are a little blurry, as a result to span a pretty broad range of the known viral phylogeny. To give you a little bit of a sense of how some of the underlying algorithms work, in a profiling tool like Metaflan, we identify gene families that are unique to each taxon of interest. So gene families that every bug in a particular bacterial species has and that no other bacterial species have. That's basically impossible for viral genes, but we can use the same concept to find portions of viral genomes that are more or less universal among a particular taxon and not carried by other viruses. So we do some initial clustering at a whole genome length, but then we shear cluster representatives into short nucleotide windows so that we can match those windows across all against all matching, eliminate windows that are not unique to each taxon of interest, and then merge adjacent windows into markers. So for anyone who's into the bioinformatics and familiar with some of the profiling methods like MOTUs, like Metaflan, this is sort of a virally targeted adaptation of those algorithms. And it lets us create surprisingly specific, unique nucleotide marker sequences for viruses. We have a slightly different pipeline that's used for protein matching. As a quick demonstration that this actually works, it's quite interesting trying to identify gold standard viruses to make synthetic data out of. What we ended up doing is splitting the viral world into known taxa that are within the expected species to genus level bounds of a VGB that's in our database. Novel taxa that have been captured by VMAG and other databases since the time at which we created our database. So these are viruses that we should not recognize. However, because viral genomes are so squishy, there's a gray area between those two areas, two genomic similarity regions that we'll call decoy viruses. These are close enough to known viral taxa that they could legitimately be part of them, but we're not sure. And based on the definitions of those taxa phylogenetically, we can't really be sure yet. So we've created synthetic data to make sure that baklava is working that specifically looks at all three of these regions. So known viruses we should always find with high precision, high recall. Novel viruses we should not find. We want low recall. And if we don't find them, then precision doesn't mean much. decoy viruses, you can make a philosophical argument both ways. So we're including them to kind of see what's happening. Fortunately, and this shows results combining nucleotide and protein level matching, we do see roughly what we would expect, where for both even and staggered synthetic communities, where we combine several viral taxa that we know by definition what's in the community, we get very high recall for the red dots, which are the known virus, very high precision for the red dots, moderate recall and precision for the decoy viruses, where again, you can make an argument that you would want these either very high or very low, depending on what you believe about viral taxonomy. And fortunately, very low recall for the blue dots, where these are viruses that we should not incorrectly call as present. So this is just a quick overview, both of the methods and some data showing that it's working so far. We do have lots of other evaluations of this. But I want to make sure there's time today to talk about some of the early screens we've been able to use this for to uncover better ideas of whole virome community profiling in conditions of population level interest like inflammatory bowel disease. So the first large-scale test data set that we've been looking into for this is the Human Microbiome Project's second wave of data, or HMP2, in which we and a couple of other teams looked at longitudinal multi-omic data from a collection of inflammatory bowel disease patients and accompanying controls. So if you see the IBD-MDB acronym, that's for the Inflammatory Bowel Disease Multi-Omics Database. It's the set of longitudinal multi-omics data that we built as part of the HMP2, capturing roughly half Crohn's disease patients, a quarter ulcerative colitis patients, a quarter non-IBD controls, for more or less 1 year each. And we have stool samples that we're focusing on for virome profiling, but a variety of other biospecimens and data types as well. One of the nice things here, though, is that it does give us paired DNA and RNA metagenomes and metatranscriptomes to look at both DNA and RNA viruses. One of the nice things that we can see that differs from the couple of other previous IBD virome studies is that much of the viral ecology does parallel that of the bacterial portion of the microbiome. There was some question in previous studies whether that was actually true, since you get different answers depending on which subset of the virome you focus on. When we see something approaching a global picture of the stool virome in this case, we see a decrease in virome diversity even just at disease diagnosis, which this is not true for the microbiome overall. You see relatively little dysbiosis or perturbation in disease that is not active yet. We see that decrease in diversity in the virome even prior to disease activity. We see a greater decrease during disease activity, and this is in line with what we'd expect from the bacterial component of the microbiome. And IBD, as an inflammatory condition, destabilizes virome composition overall. So if we look at the amount of diversity between individuals, either across all samples or those that are not dysbiotic, either control individuals or IBD patients currently with currently quiescent disease, these retain kind of a smaller radius. They're closer to the homeostatic area of the ecology, which starts to be lost either among individuals with active disease who are in dysbiosis or when comparing those to control individuals. Even despite this destabilization, both within IBD patients and controls, viromes are as much or more personalized than the bacterial component of the microbiome. So within individual, there's a great deal of retention of the viruses that you have over time. This is quite similar to, say, microbial strain genetics, which tend to be retained within individual over time. And like those bacterial strain genetics, there are huge differences in which viruses are carried among individuals. And this is in line with some of the other population virome studies that have been carried out previously in other phenotypes. One thing that we thought was interesting here is the way in which prevalent viruses are distributed among individuals. Some individuals, internally persistent viromes, are made up mostly of viruses that we also see in other individuals. So those would be the ones in dark blue over on the left here. Some individuals' viromes tend to be internally consistent, so they're maintained within individual over time, but the viruses that that individual maintains are quite different from any of the other individuals in this population. And there's really quite a continuum between individuals who tend to carry viruses that are similar to everyone else's over time and individuals who tend to carry really distinct their own viruses over time. Despite this persistence of personalized viromes, this is another thing that IBD destabilizes. So Crohn's and colitis patients tend to lose more of those personalized viromes gradually over time. So within subject, the longer of a time period over the course of about 1 year per individual, the fraction of the virome that tends to be gradually lost, and that fraction is both accelerated and greater among ulcerative colitis and Crohn's disease patients. And we can see this either by counting the number of maintained viruses per individual or the fraction of maintained viruses per individual. Most individuals, though, even those with IBD, tend to be up in the very high fraction of viral abundance that is maintained over time. So, like bacterial components of the microbiome, there's a fringe of less abundant viruses that are turning over much more quickly than the abundant core viruses per individual. And I mentioned earlier that the IBD patients start out with a smaller number of viruses, a lower viral diversity to begin with. Like other microbial community profiles, we have some statistical methods that can explicitly associate features of a microbial community with phenotypes and exposures and covariates. So we've previously published as part of the HMP2 portions of the microbiome that associate with IBD diagnosis, with dysbiosis, with other exposures that go along with IBD, like antibiotic usage. So we can do that again, both using more recent profiling of the HMP2 data and using an updated version of our statistical package for doing this, Maslin. And I will advertise it today, but not really say much about it, except in as much as the version 3 that we've just recently finished includes some new statistical tricks, some additional experimental design types that can be modeled. And especially if you notice here, we'll separate associations between microbial community members based on their presence and absence or their prevalence versus associations that are based on their abundance. We've known for a while now that microbially associated phenotypes can be caused by both things in different circumstances. Sometimes the abundance of the bug matters, sometimes the presence of the bug matters, and now we explicitly model both of those things. So we can do that for bacteria, which is quite similar to what we previously published, but we can now also do this for viral profiles as well. If we put together a statistical model that's basically the same, it's exactly the same as what we've used previously for bacterial community members, we can capture the effect of disease diagnosis, disease activity, other exposures or demographics per individual, and the random effect, the longitudinal effect of multiple samples per participant to identify which viruses specifically are disrupted during IBD diagnosis or activity. Many of these, like every profile that we've looked at so far, tend to be viruses that we can't even give a speculative taxonomy to yet. Many of them tend to be abundant gut phage, like grass phage as one of the most common examples. And like bacteria, like I mentioned the reduced diversity earlier, viruses tend to be lost during IBD. There are almost none that are gained or more abundant. Hot off the press, we can do the same thing for RNA viruses, but all I know so far is that we have data on it. We just barely finished these profiles. So we again see a really interesting combination of uncharacterized RNA viruses, DNA viral transcripts, and then we've clipped off here many of the RNA viruses that have known to be part of the gut virome for quite a while are plant viruses. So all of the tomato mottle virus and pepper mottle virus, would scroll off the bottom of the slide here. So stay tuned for some additional actual biology and epidemiology on that, but we at least have the data now. In the last bit of the talk here, I wanted to speak to another acronym, a completely different project that is ongoing. We've recently finished some aspects of it, though, that I think are just fun to talk about, and that's the One Health Microbiome Resource, or OHMR. This is a combination of a really fun industry collaboration, a data resource that's available to the community, and some individually interesting biological studies that we've been able to do, all in collaboration with Hill's Pet Nutrition. And so this is a group that I never expected to work with at a school of public health. But what I found out from one of their scientists that I met a few years ago is that they have this fantastic research facility in Kansas that's puppy and kitty heaven. There's about not quite 1,000 dogs and 400 or so cats who live most of their lives there. Their whole job is to eat different food and see if they like it or not. It's fantastic, and we get way better data from these animals than we ever could from human nutritional epidemiology. So it's been just delightful to work with the team. And one of the things that we were able to do is that they had thousands of internal shotgun metagenomes from different studies in these animals. That's about 4 times more data than was ever previously published for canine and feline gut microbiomes. So we put together a data resource to share that, again, with the community, the OHMR, One Health Microbiome resource. That's online now with a searchable index of both the Hills-generated data, and we've integrated that with the dozen or so previously published canine and feline shotgun metagenomes. As an overview of the data contained there, we did sort of a high-level meta-analysis of roughly 2,000 canine gut shotgun metagenomes, 300 or 400 feline, and then we integrated that with several hundred metagenomes from the HMP1 and 100, 150 metagenomes from an external Malagasy population, kind of as a human outgroup. Of these total 2,500 or so samples, around 2,000 come from Hills and our new data sets. Around 500 are previously published. So we ran these through a consistent pipeline based on our biobakery workflows, where we assemble and profile each sample in the same way. We quality control them, so on and so on. And this allows us both to make them available and to do some high-level statistics on what kinds of microbial profiles or microbial genetics are different between human microbial lineages and animal, or shared within different companion animals. And of course, at a high level, like many of the mostly 16S-based studies that have been published before, the overall microbial taxonomic profiles are quite different. So if we just look at a high-level ordination of a few hundred humans in blue, a few hundred cats in red, and a few thousand dogs in yellow, yep, taxonomically, everything is quite different. This is based on Bray-Curtis dissimilarity. If we do the same thing using something like weighted unifrac instead, much greater collapse. So different specific bugs, very phylogenetically similar bugs. So I like to think, and the same is true in lab mice, for example. All of the bugs tend to be a little different than what we see in the human microbiome, but they're cousins. They're very consistently closely related. So you can use this to do similar things as would be done with 16S profiles. We can find specific bugs that are carried by all 3 hosts. So some of those are of particular epidemiological interest, like Prevotella capri, we know has kind of a unique functional and ecological role in the human gut. It's one of the few bugs that's abundant in people and cats and dogs. We can find plenty of both characterized known species level genome bins, and this is going back to bacterial profiling here, and novel or uncharacterized SGBs that are distinct from existing isolates. And of course, we can find many of both known and unknown or novel SGBs that are unique to humans or shared among companion animals or unique to individual hosts. And so all of this isn't too surprising, except maybe some of these strong overlaps. But what's really fun about the shotgun metagenomic data is that we can break this down to really detailed strain genetics and ask what the expected transmission or evolutionary histories of these shared or distinct bugs has been. And again, a method I won't really go into much today that we've developed to do this kind of thing is called ANPAN, which is specifically for strain level genetic epidemiology from microbial communities. Very briefly, ANPAN will start from QC'd taxonomic and functional profiles. First, identify which bugs in which samples can be analyzed at the strain level, which ones have enough data, enough sequencing depth to make that feasible. And then ANPAN supports 3 different types of statistical models for this, one of which looks at strain-specific gene carriage. So are there genes that are gained or lost in strains that are specific to a particular environment, or in this case, host? One model looks at strain phylogeny. So are there evolutionary patterns that are non-randomly distributed among different environments or in this case, hosts? And then a third model that looks at pathway gain and loss or pathway persistence within strains that are specific to a particular environment. And we'll focus just on this middle model for today, looking at some of the phylogenetic patterns among bugs that are nominally the same between all three of our hosts, humans and cats and dogs. So if we use this more detailed view to answer how many bugs are you sharing with your pets, or how many bugs have been historically shared among humans and their companion animals, you get a somewhat different answer than the high-level view might show. So if we take an example like what used to be B. vulgatus, currently Phocacola vulgatus, this is a bug where historically there looks to have been many, many transmission events between different hosts. So again, blue here is people, red is cats, yellow is dogs. And you can see that the tree is extremely intercalated. And each of those branch points represents a predicted transmission event between different hosts. So evolutionarily, historically, there's been a lot of transmission of this bug. It doesn't really care so much which host it lives on. But you can also, like most things in biology, you can also find exactly the opposite answer. If I take this second bug, Blauschia, and look at its evolutionary history, it seems like it's very rarely transfers between hosts. There was one historical transmission event between dogs and cats and dogs and humans, and that's it. So this bug has essentially subspeciated into lineages that are specific to a particular host, and they don't like to go from a person to a dog or vice versa and so on. And you can find intermediate cases as well, where something like Ruminococcus navis, which is a bug that's particularly of interest in IBD and other inflammatory conditions, this is a bug that tends to be host-specific, but occasionally transmits. And this is even more interesting because it's a fairly rare bug in humans. It's only typically carried or abundant during GI inflammation, very prevalent in dogs. Often with different genetics, we don't really know the reasons for this yet, but there could be something ecologically and epidemiologically important about these rare transmission events between humans and companion animals in this case. And you see there's one historical transmission into cats. And if you squint at some of these tips, there are occasionally additional recent transmission events, generally here from dogs to people. So we can quantify this. We can take these kind of intuitions about the phylogenies and lay them out on a spectrum from frequently transmitted and thus shared microbes and infrequently transmitted or host-specific lineages. And like I mentioned, there's lots of ongoing work here too that I don't have time to get into today, and specific studies on fiber intake, low fiber diets, early life development, where you can again get very specific and more detailed data than a lot of human studies, antibiotic exposure, where these animals have very rare but very controlled antibiotics exposures when a puppy gets injured on the playground or whatnot. And we can deep dive some of the metabolism here because we get more parallel multiomics data. Because very fresh samples can be collected, we can get DNA and RNA and metabolomics and serum metabolomics and so on from many of these studies. I got my 1 minute warning, so I'll wrap up with one more advertisement for our Harvard Chan Microbiome in Public Health Microbiome Center. Parallels a lot of the activity that's gone on here. I'm very excited to talk to some of the center members here later on about similarities and differences. We have a lot of similar initiatives to help with research, education, outreach. Some activities that may be of interest here, we have a very large microbiome collection core that helps with human microbiome population studies. So if you don't already have the capacity here and you want to collect tens of thousands of stool samples, let us know. That's a thing we can do. A similar bioinformatics core, but I particularly want to highlight our annual symposiums coming up in a few months. It's also a hybrid format, so feel free to join us on either in person in Boston or online. And for the first time this year, we're also offering a week-long short course to be hosted at the Broad in Boston about a couple weeks later. So if you're interested in either of these, take a look at the information for the center, the symposium, and the course online. And I want to thank again everyone whose pictures you saw are either people from the lab who did all of this work or are fantastic collaborators externally or within the HCMPH in Boston. Thanks so much.
Well, thank you so much. It's really a joy to be here. In some ways, it's like old homecoming, seeing good friends that we haven't seen for a while. Um, yeah, so I'll just jump into this. I kind of struggled with the title, so this is what we have for right now. And I feel like we're going to take a hard left turn from all of the great stuff that we heard from Curtis just earlier. So my world is in the environmental sciences, and I'm always really impressed to hear about the latest and greatest research and gut microbiome, human microbiome, because I feel like we just don't necessarily have the same tools that we can apply to different environmental samples, and just very envious of some of the resources. So I wanted to start today with something that has really interested me for several years now at the JGI, and that's soil environment. My background is in oceanography, and you can just go out to a glamorous site on a boat, filter water, and it's just very easy to deal with. And when I joined the JGI way back in the day, we got most of our proposals from the community, from soil scientists, and it has just really baffled me in terms of there's been so much research done over many, many decades in soil sciences, some great molecular tools that have been applied to try to crack open the microbial dark matter within various systems. And then, over the holidays, we try to do some fun activities with the kids. And we have this little terrarium that my 5-year-old is holding up, and just some basic questions that a kindergartner asks that, we maybe still don't really know. Why is soil stratified the way it is? How are these little grasses and alfalfa seeds growing, and what are the connections with the different types of soils? Why can't we just go out and grab some soil from our backyard? Why does it need to come in this, little kit? All fun questions that, as a scientist, I think, yeah, I have that same type of curiosity. So that's where I wanted to start. And my world over at the JGI, which is just across the bay, I think we have a lot of the same parking troubles that UCSF has here, but otherwise, could be considered very different worlds in the microbiome space. And a lot of the work that we do at the JGI is focused on kind of this pipeline here, where we do a lot of shotgun sequencing from really diverse samples around the world. So anything that doesn't have a vertebrate, soil, ocean water. We generate a lot of short-read shotgun sequencing, and then we try to reconstruct those population genomes using fairly standard automated scripts, and then we try to say something about the community function and the metabolism. And I'm sorry today if you were interested in hearing about all of the great mags that we've generated. We are in the process of doing a lot more. Several years ago, we did big study, a lot of researchers involved to try to better catalog the mags across the globe. We're in the process of doing that, but today I wanted to maybe focus on a nagging problem that's been really bothering me and folks in my group for a while, is that when you do mags, you only really capture a very small amount of data that you actually sequence. And this is from one of the studies we came out with, led by Stephen Nyfox several years ago, where we did a whole bunch of binning. We were really excited. We increased the amount of phylogenomic diversity that had been cataloged, and that was the main emphasis of the study. But then, kind of hidden in the supplementary material was this figure, which we ended up mapping the unassembled reads to this catalog on top of, at the time, the RefSeq catalog. And it was actually pretty disappointing to see this. We highlighted that we had a 3.6-fold increase in the number of reads that could be mapped. But when we dug down and looked at the data, really about 70% of the reads still couldn't be mapped to anything in the reference catalogs. And this has been something that, continues to persist. There's been lots of new MAG catalogs that have come out that are very impressive, that are looking at new environments. But the fact is that for shotgun metagenomics, there's still a large portion of reads that can't be mapped. And so one very talented scientist in the group, Matt Kellum, decided to look at shotgun sequence data from a different perspective. And as a geochemist, he had a lot of training in this idea of a phase diagram and borrowing that concept. So within this phase diagram, looking at the chemical composition of different minerals and how they display out. And so Matt wanted to look at this idea and apply this idea to shotgun metagenomics and try to better capture what are, from a community-based perspective, what are the different ways that the environment impacts those microbiomes across many different environments. And so what he set out to do, and this is a concept that actually Forrest Rower had done many, many years ago, looking at virome sequence, was to see if there were genomic signatures that could be used to compare metagenomes to one another. Now, this isn't using tetranucleotide frequencies, which are commonly used to bin metagenomes, but this is an approach to compare different metagenomes to one another. And so when we think about taking this phase diagram analogy, we are taking the tetranucleotide frequencies, or the KMERs, and treating them as the quote unquote chemical composition within a metagenome. And so for those of you who just need a refresher, you break out the tetranucleotide frequencies that's shown on the right-hand side there. And then, of course, we need to make some assumptions. And these are that closely related taxa share similar frequencies, and that presumably metagenomes share from similar environments have similar frequencies. And so at the JGI, we're very fortunate to have some really nicely curated databases that we work with. One is called the Genomes Online Database, or GOLD database, that has a team of curators to classify metagenomes that come through the JGI, either through our proposal mechanism or are submitted directly by users who are outside of the JGI. And this is what the GOLD classification system looks like. There's other classification systems out there depending on what reference database you have, with different flavors of how you can describe different environments. But for our purposes, this is how GOLD describes the 5 different level categories. And then we also have the IMG database, the Integrated Microbial Genomes and Microbiomes database that hosts many, many thousands of public metagenomes, again, both from the JGI as well as externally submitted data sets. And we were able to pull 12,000 public metagenomes for this initial analysis, with the different ecosystems shown there. And, just comment, of course, this is predominantly non-human microbiomes. So we're talking only about environmental samples here, but of course, biases in environmental sampling as well. Okay, so once we gathered that data, the process is as follows. So Matt broke down the tetranucleotide features across all of these 12,000 metagenomes. And then to be able to analyze that data, he used LDA, or linear discriminant analysis plotting, to reduce the dimensionality of all of those frequencies across all of those metagenomes. And then to further use classification, he used K nearest means classification using those different ecosystem classifications. And then one of the nice features, because we're only talking at this stage about raw reads, that we could use the annotations from those assembled metagenomes to overlay the gene information from those metagenomes. And so we're combining a reference-free approach with the annotated information that had also been alongside how we process these metagenomes. And so we could overlay the relative gene abundance normalized based on the size of the metagenome. Okay, so what does this look like? It looks like this. And we were really interested in digging into this data. This is not only was this a way to analyze metagenomes separate from how we traditionally have been analyzing using a more genome resolved approach, but we've also, Matt's a very good coder, and he developed this as an interactive tool. And so I'll get to that in a minute. But what we see is, what you would likely expect based on our assumptions, that microbiome composition across unique ecosystems partition out. So that this is not anything unexpected. You see marine up in green, non-marine saline and alkaline, close to freshwater, soils up there, and then thermal springs. Those communities are, just very different in terms of community composition from everything else. And so if you zoom into this plot, so each little dot is an individual metagenome profile. And I know that it's probably hard to see. Again, it would be nice to have kind of more of a visual, but this is an interactive plot where you can identify which metagenome it is and what the classification is. So it's a data discovery tool to be able to identify where your metagenome compares against about 12,000 other metagenomes. Okay, so we have this nice interactive tool and visualization. One of the first things that we were interested in asking was, are there specific functions, annotations, gene annotations that differentiate across ecosystems? And this has been done before from a different, using different approaches, using annotated genes, as opposed to using the reference-free approach that we had done using this tetranucleotide based approach. And so I'm going to take you through one example. And again, the coloring here, so there's the overlay of the ecosystems, and then this normalized gene abundance bar down here is an example of one gene that was identified out of a very long list of ranked genes that differentiates across different types of ecosystems. And in this example, this gene, COG3237, unfortunately an uncharacterized protein, was one of the most differentiated across ecosystems and specific to soil. And so you see over here, higher abundance. Matt and I had a lot of exchanges over should dark be high or low. So this is how he plotted it. So the yellow is low abundance and the dark is high abundance. And so this is, again, one way to analyze the data, to look at how functions can differentiate across ecosystems. And in this case, we did a little bit more digging. There's not much you can say about uncharacterized proteins, but there were some literature citations that maybe this could be involved in osmotic stress or desiccation. So you can make some hand-wavy things about this as you like. Another way to look at this is not within the plot, but when you look at the abundance, the normalized abundance, you can see that there is a significant amount in terrestrial and soil environments compared to everything else that's compared. All right, another way to look at this data and use this phase diagram tool is to think about nutrient cycling across ecosystem. And that's a lot of what we do at the JGI, thinking about biogeochemical cycles and how they interface across environments. So Matt has been very interested in the nitrogen cycle for a while. Before coming to the JGI, he was working on ammonia oxidizing archaea. And so he wanted to look at a couple examples of nitrogen cycling genes across environments. And so what this looked like for one of the key genes for the anammox near K, that there was an overrepresentation in soil environments. Similarly, the assimilatory nitrite reductase, NIRB, which is taking nitrite to ammonium, was also overrepresented in soil environments. And then contrast to that is the dissimilatory marker genes being overexpressed or overabundant in terrestrial systems. And so again, this is one way to take this type of analysis and look for different pathways that might be of interest from a biogeochemicals perspective. And so the preprint is posted with QR codes. For those who know me, I love QR codes. Is available if you're interested. If you go to the website right now, unfortunately, we are having a maintenance, so it's not available, but I would encourage next week, if you're interested, to go in. You can go in either through IMG or through microbiomes.jgi.doe.gov to play around. And again, data discovery tool. We plan to make annual updates. Right now, we only have 12,000 metagenomes. Probably in a year's time, we'll have many thousand more. And we welcome input in terms of if this is a valuable way to analyze environmental data. All right, so I wanted to next transition to, that's just 12,000 metagenomes. And we heard from Curtis earlier, and we know that there are many, many thousands more metagenomes that have been generated and are publicly available. So when you do a search at NCBI, you get about 500,000 metagenomes that are publicly available. And so wouldn't it be nice to improve at least the tool that I just showed, as well as additional analyses in terms of having a lot more data. One of the other hats that I wear outside of JGI is being a part of the National Microbiome Data Collaborative, which is an effort to democratize data science for microbial research. And one of the things that I've been very, very excited about over the past year is providing new tools and resources to improve metadata capture and collection across diverse samples. For those of you who may or may not work with the JGI, you might be familiar that we email spreadsheets back and forth, which I personally believe is a little bit painful and prone to a lot of human error. So over the past year, the NMDC team has been working on developing a mobile app to collect field measurements and sample information directly in a standardized and simplified way. There's been other efforts and tools to have similar types of resources may be project specific. This is a generalized tool based on the Genomic Standards Consortium MICSES standards, and it's fully compliant with those standards, as well as a process that we have for capturing this information and then pushing data directly to NCBI so that it's fully compliant with standards. We've been in the beta testing phase for the past year. So if this is of interest, we would welcome feedback from anyone who wants to test it out. But I'm super excited that we're launching this in April. It will be available in the Apple and Google Play stores for anyone, anywhere, to get access to and play around with. So we're hoping that this will be one way that we can avoid sharing spreadsheets back and forth, and will hopefully help with the data management process going forward. All right, and then in the last couple minutes, I wanted to just share a newer effort that we've been working on that I, again, think will be a really exciting path towards improving how microbiome scientists can manage their data and report on their data in a more systematic way. I don't know how it is in your world, but at least in the Department of Energy world, all of the talk is about AI and how AI will solve a lot of these problems that researchers are facing. I don't know if I believe that, but I do believe that if we improve how we report on the data that we generate and the study information that we have, that we can get us maybe a little bit closer to ways that will improve, some future AI world. So a couple years ago, we reached out to Chloe and Levi, who were the architects, along with Curtis, for developing the storms reporting guidelines. They were very interested in the idea of improving storms for the environmental sciences. And after a couple discussions, we just said, this could go along very well with just adopting what you've already developed for environmental sciences, non-human host, and synthetic microbiomes. So last summer, prior to the ASM microbe meeting, we had a data management in action workshop. It was a really great workshop. It was led by Julia Kelleher, who's just a force to be reckoned with, along with Lynn, Chloe, Scott from NIST, and Zoe, who really helped with all of the logistics and management. And very briefly, if you're interested, the workshop report has been posted on Zenodo, but we spent the first day talking about, a lot of the things that we've been talking about for decades now is, we have all of this data, what are we going to do? Oh, you work in this system, I work in this system, they're very similar. What was different about this workshop is that we took representatives from many of the main funding agencies within the US government, many of the main publishers who publish on microbiome research, and then research panels and data repository representatives, and talked about what we can actually do beyond just putting out a paper with another set of reporting guidelines. And so from storms to streams, this is Julia's idea with acronyms. You can talk to her about that if you want. But following the workshop, we've been having bi-weekly meetings. They're open meetings, so if you're interested in joining, that would be really great. We've already had 2 rounds of feedback with over 1,000 comments from about 250 researchers in 30 countries. We also have volunteers to do different case studies of how this would work in practice. Given the past 2 weeks of kind of recent events, we're on pause right now because we have a lot of federal colleagues who are not able to directly work on this. So I would invite anyone who's interested in doing this to join, because now is a great time to hop on in. And we will be socializing this with publishers and funding agencies for adoption going forward. All right, so I think I'm at time. I have a great team at the JGI and at NMDC, and I'm happy to take any questions.
Well, thank you so much for a great invitation. I'm really happy to be here. And I just think it's amazing that most of the microbes that live in our bodies are uninvited guests, because this is not something we're really controlling. This is something that's just happening on its own. And for the most part, it's going really well. But of course, if you get the wrong guest in the wrong place at the wrong time, it can cause a lot of trouble. And so I'm excited to tell you guys about some of the projects I've been doing through the years focused on oral microbiomes, which are some of the densest and most diverse communities on the planet, and how that can sometimes impact our airway health as well, especially in the context of cystic fibrosis. So I'm joining you from SoCal. I'm a Northern Californian myself, actually, but soon it'll be longer that I've lived in Southern California. And we have beaches and mountains in SoCal too. So I've been at UCI. And so I've been at UCI. I've had a lab there for 10 years now. And I also just want to point out that we have a SoCal Microbiome Symposium every fall, and maybe it's a little bit tentative, but the plan is to host it at Caltech this year. So maybe I'll get to see some of you there as well. And before I get too far, I just want to really acknowledge the team of people I've gotten to work with, collaborators and students and postdocs in our group. And we have a really great microbiome community at UCI, so it's been a really nice place to work. I also really care about these standards efforts. If we're going to call a microbiome, we better be using the same rulers. And we have this goal as part of the Microbiome Centers Consortia Standards Committee and the IMSA, which is part of NIST, to develop a top 10 guiding principles document for microbial standards. So if anybody has any ideas that way, I'd be interested to hear them. Okay, so today I want to focus on data that helps us understand polymicrobial challenges of our time. And so I would argue that these are big challenges where having an understanding of a microbial community as a whole is going to really help us make progress. So if we just try to look at one microbe at a time, we're not going to be able to understand the whole system. And some of these are really enormous problems. And I would argue that neurodegenerative diseases and autoimmune diseases might belong on this list as well. I just recently updated the number about the size of the probiotic industry, $80 billion globally. And there's just so much potential there to make that turn into something effective, I think. I don't need to introduce the microbiome to this room, but as you know, most of the genetic differences between you and anyone else in this room are encoded in our microbes. And a lot of them are actually in our oral cavity. I think we think about the gut a lot more. And so if you look here, colon and dental plaque are some of the densest microbial communities that we have. And then of course, saliva is more of a complex mix of all of the different oral microbial communities. I also think it's amazing that we have so many anaerobes living in our mouth. I remember telling that to dentists when I first started working on oral microbiomes, and I heard this kind of gasp in the room that you could have so many bacteria that couldn't thrive with the sight of a molecule of oxygen, and yet there they are, just right on the surface of our teeth. So we have tons of anaerobes in our mouths, lots of streptococcus. They're super unique to each person, but you see similarities in households and families. And there's big oral and systemic health impacts of the oral microbiome, of course, too. So my first microbiome project ever was an oral microbiome project. I think it took me more than a month to install Chime at that time. It wasn't a nice little package at that time, but it was such a fun time to be a microbiome scientist. There was one paper a month. I could read everything. And and this was my bike ride. I was a postdoc at the University of Geneva Hospitals in Switzerland, and we started just collecting saliva samples from healthy people, and we didn't know anything, like what was going to be in there. Did people have a consistent microbiome that was stable through time? And of course they did. So we found that the saliva community composition was actually quite stable in people through time. And that was really interesting. And then I had a really unique opportunity to study children who are affected by a disease I had never heard of, but it's actually affecting many hundreds of thousands of people around the world in the context of malnourishment. It's still a big problem, especially in Africa. And so we had no idea what the etiology of NOMA was. And I had the opportunity to study samples from wound sites where this terrible facial gangrene erupts in children who develop NOMA, along with healthy sites from the same mouths. And we had controls from the same villages. And many of the etiologic bacteria that were considered to be causative of NOMA were very normal in the healthy controls from those villages. So it made the context of what is healthy so real to me. And just the idea of having household controls and environmental controls, that was something that came really through in this project for me. And there's a new effort. One reason I included this, just one slide about this study, is that there's a new effort, the International NOMA Network. And I just wanted to broadcast that because there's a lot of knowledge in this room and people might have ideas for how to do a better job of studying this. On a totally much bigger and more modern scale, I've also gotten to work on some really big saliva microbiome projects as part of the NIH ECHO cohort, the Family Life Project. And now we've published a couple papers about it, and I'm just highlighting here that when you get 1,000 samples from mothers and their kids, mostly mothers, her caregivers and their kids, you see really strong family connection in both the microbiome and the metabolome. And we didn't actually see big signals from the very rich metadata that we had in these samples. People tend to cluster into one of two clusters for the microbiome and also for the metabolome. And that's the biggest signal in our data aside from the household that the sample came from. Okay, I'm just kind of giving you some highlights here. I want to really dig into an unpublished data set that I had a very unique opportunity to work with an anthropologist at Cal State Fullerton, who has one of the longest field sites studies going in the Amazon in Ecuador. And my grad student, Andrew, I have to really showcase his efforts here because he did a great job with all of this. He studied undergrads at UCI and also people in the Amazon, which gave him a really unique perspective on saliva microbiomes, that's for sure. So here's the team down in the Amazon. They went one summer and collected a huge number of samples, and it was a really hard project. There's no electricity, so we had to be really careful in how we collected and stored the samples, and they just did a great job. There were a team of master's students who are also depicted here, who I have to thank for collecting these samples. So the people in the village we studied, Canambo in Ecuador, get most of their calories from a beverage called chicha, which is a fermented root vegetable beverage that the women of the household make by chewing yucca roots, which are depicted here. And they spit it into a vat and they let it ferment for a couple of days, and it gets a couple of % alcohol, and then everybody drinks it, children included. And it's, really keeping them healthy. I've heard they might be getting as many as 3,000 calories per day from drinking chicha. And of course, it's very connected to the oral microbiome because that's what's seeding the beginning of the fermentation. The people tend to be really healthy in this village. There's skin health is just amazing. There's no acne or eczema or anything like that. And so there are just a lot to learn about the diet in that place. Like, for example, apparently the kids love taking grubs that grow on rotting vegetables and smoking them up, like getting them a little toasty. And that's like the best snack ever, apparently. So we got to ask questions about what microbial taxa are present in the people of Canambo. We collected fecal, saliva, and chicha samples. And for today's talk, I'm going to focus on the saliva and the chicha samples, and I'm happy to talk about the gut ones too. So here's just a small corner of the sample collection effort that you can see here. So we got all these samples coming back. They were collected in Zymo buffer, so we didn't have to refrigerate them, so we couldn't do metabolomics. So just big picture, what taxa were present in these samples. And, you'll be happy to see that the saliva and the fecal and the chicha samples each have very distinct microbial community composition. But we see some really high abundance of bacteria that are vanishing from the industrialized world, like Prevotella, for example, was extremely abundant in the fecal samples. But I've been promising you here that we're going to focus on the saliva and the chicha samples, and you'll see that they're more connected in terms of their composition in some ways. But we see tons of lactobacillus in the chicha samples, and you'll see some other well-known oral bugs are quite abundant in the saliva samples. We did find that cohabitation has a big influence on the microbiome composition. And here I'm showing you the Bray-Curtis distances, where you can see that samples from within the same household have more similar chicha microbiomes. And that's also true, maybe even more so for the saliva samples than for the fecal samples on this Bray-Curtis distance metric. So the saliva samples were a good signature of household, and that extended to the chicha samples as well. If you look more comprehensively at the composition of the oral microbiome, this is shotgun data, so we can see fungi and viruses to some extent as well. And you see that the majority of the microbiome comes from Streptococcus salivarius specifically, and that's exactly what you see in the industrialized world too. So maybe there's less of a big difference in the oral microbiome composition compared to what we see in gut microbiome studies. That was true for our data as well. But we also found that huge numbers of the reads were unknowns. And that's actually getting better. Like I wish I had the Metaflan 4 data up here, because I do have it, and the many more reads mapped with Metaflan 4. So it got a lot better, I have to say. But still, there's tons of unknowns. When you assemble things, same thing. Most of the reads are not getting into the assemblies. So it just is a reminder that there's so much genomic dark matter, and I love these database independent approaches. We also tried K-mer analysis in this study, and it captured a lot more diversity and was really fun also just to get to see all that data getting included. So another question you might have is like, okay, so given a saliva sample with all those teeming millions of cells of microbes in there, which ones actually inoculate the chicha? And so we got some time points from 4 days of fermentation from the chicha. And you can see that they start out, the Streptococcus is quite important, rearing out the gate. But then lactobacillus takes over, and there's always a bit of Saccharomyces involved as well. So that was a really interesting thing to get to see. A lot of people were curious about how important the saliva microbes were for seeding the chicha fermentation. We also looked at how household specific the Streptococcus were, because the chicha is made at a household level. So it's interesting to know how different the strains are from different households. And we definitely see that there's a household signature for the Streptococcus strains in these samples. We also had a really interesting control, because the anthropology students who were there for the summer, they also fermented chicha. So we got to study their Streptococcus strains, and they had very distinct Streptococcus strains in comparison to the people from Kanambo. So, okay, I'm going to summarize my microbiomes from Kanambo project. And I think a major thing to say is that the Streptococcus were indeed quite household specific, play an important role in the early chicha fermentation, and just that there were a lot of unknowns, which we're doing our best to capture using database independent approaches. So that brings me to talking about the ecology of airway infections. And, still today, the most common way that an airway infection is probed in a hospital is by taking a swab or some type of sample, a sputum sample, if you can get one, and then cultivating individual pathogens in the clinical microlab. Typically, if one of the bugs that lives in the oral cavity cultures out, it's thrown away and considered normal flora and unimportant to the infection. But we actually know that a lot of even the pathogens that do end up colonizing the lungs, they often can't do that by themselves. For example, Pseudomonas, a really common pathogen in cystic fibrosis infections, it can't ferment mucus on its own, but it can eat the fermentation products of Streptococcus fermenting the mucus. So there's a lot of really interesting and important exchange, and these pathogens are not arriving at the lungs alone. So I like to look at these as two communities that are interacting in the context of an infection. And they don't even have to be right next to each other for this interaction to be important. For example, Streptococcus could be happily buzzing away, fermenting volatile molecules, and a Pseudomonas cell quite distal in the lung could be fed off of volatile molecules that are traveling from that fermentation. So this is something that I was really interested in as I was first starting my lab. I was taking breath samples from people at the clinic in San Diego and studying the fermentation products emerging from breath samples from people with cystic fibrosis. And we definitely could find specific molecules. We could figure out which bacteria were producing them. One of my favorites, 2,3-butane-dione, is the same molecule that's used to flavor microwave popcorn, and it's quite toxic. So we were really excited about using these molecules as biomarkers to try to detect infections earlier or to try to help treat them more specifically, and so on. So, and actually a really important thing to say, and this is really good news, we could all use some good news right now. Maybe many of you are familiar with this, but about 5 years ago, there became new drugs available for people with cystic fibrosis. They're channel modifying drugs that actually address the cause of the problem in cystic fibrosis. They help the transmembrane proteins that are misfolded or not working correctly actually function. And so this whole community is just in a very different situation now. It's wonderful. The life expectancy has increased 20 years or more. It used to be something like in the 30s, and now it's like in the 60s. And just quality of life is much better. Importantly, collecting a sputum sample, which was the basis of most of the microbiology for studying cystic fibrosis, is not even possible anymore because people are so much healthier, which is obviously really, really good news. But it makes this study that I'm about to tell you about kind of special because we have tons and tons of sputum samples, which will never happen again. So I had a really lucky collaboration with John LaPuma to study the STOP2 trial, which was carried out by the Cystic Fibrosis Foundation. And I have a bunch of data to share with you today, looking at the microbiomes and metabolomes from a large number of people in North America across 50 clinics in North America. So the study was set up to try to understand how long antibiotic treatment should be carried out for when people get what's called an exacerbation with cystic fibrosis, which is a period of time where your symptoms get much worse, but in some ways is defined by the doctor and the patient just deciding to diagnose it as an exacerbation in that moment. So it's a bit of a ghost to chase to really define what an exacerbation is. However, when somebody is diagnosed with an exacerbation, they often come to the hospital or somehow are given antibiotics, usually intravenously. So this study was set up to compare how long do you really need those antibiotics for, with the goal of seeing could we shorten the antibiotic treatment course and protect people from unnecessary exposure to antibiotics. And so we have nearly 1,000 samples from more than 300 people to make this comparison. And for the purposes of my data today, I'm just going to talk about visits 1, 2, and 3. I'm not going to distinguish the arms of the study, just to share a more ecological view of the data. So I have analysis from 949 sputum samples, both 16S sequencing and metabolomics. So interestingly, you can see that the microbiome diversity is pretty stable through the visits. You don't see big changes, even given this biggest hammer we have, the antibiotic treatment. You don't actually see a lot of movement in a big picture way on the microbiome side. We do see a lot of variance attributed to the individual, which makes sense. So people's communities are unique to them. If you look at the spread of the samples in terms of disease stage, you can see that there is some spread, but the clusters that are forming are not obviously clustering in terms of what disease stage the person is presenting with. If you look at diversity with disease stage, again, on the microbiome side, there's not really obvious trends. emerging, which is nice because there's been so many smaller studies. So it felt really nice to have this very big study with a lot of people and all the data collected the same way. One trend that's really clear and that we expected is that Pseudomonas, which is an important pathogen for cystic fibrosis, increases with disease stage. So that's a really clear trend, and it's nice to see it in this bigger population. If you look at the abundance of the different microbe types in the clusters that form across these microbiomes, you can see that there is actually a distinct diversity, a little bit higher diversity in the second cluster, which has more people in it. In this context, higher diversity typically is associated with health, because as the infection progresses and becomes dominated by an individual pathogen like Pseudomonas, the diversity goes down. This is not to say that we should be all like inhaling saliva microbiomes with the hope of increasing the diversity in the lungs, though. It's an interesting conundrum about how to view diversity in health in that context. So then if you look at the composition of some of the major taxa in these clusters, you'll see there's a lot more Pseudomonas in this first cluster, which includes more people with more progressed disease, whereas the cluster 2, which includes more healthy people, that one has more Staphylococcus. The Streptococcus and Prevotella patterns are quite similar to the Staphylococcus patterns. So it's interesting to see this emerging on a nice big population of people with cystic fibrosis. Then on the metabolomic side, we actually see even more of the variance is explained by the individual coming from the metabolomes. But where there wasn't significant variance attributed to the time point or the disease stage in the microbiome side, we actually do see some significance in the metabolome, which is my expectation, but potentially really useful if it means that we can have metabolomic biomarkers that help us understand disease stage or whether your antibiotic treatment is working and so forth. There's like signal in this data is what this is telling me. So if you look at the diversity during the different visits, you'll see that right after the antibiotics are given, you actually see the metabolomic diversity going up. And I'm looking at this as a reflection of cell destruction. So as the antibiotics are getting in there, they're popping both microbial cells and attracting immune cells, and that's leading to just more different types of molecules being present in the sample. If you look at the top 20 metabolites that are significantly associated with visit, as in whether the antibiotic treatment was just given, you see a lot of markers that to me are representing broken cell membranes, which was interesting for me because one of my favorite papers in this space came from a European group in 2010 where they took breath samples and compared them between people with cystic fibrosis and healthy children. And they also saw all these, molecules that to me are reflecting just broken cell membrane bits. If you look at some of the other molecules that I was really excited about, more reflecting microbial fermentation or other kinds of fermentation, you see that actually with antibiotic treatment, lactate, for example, is going down. So that's really interesting. That suggests potentially that the antibiotics are inhibiting some of the anaerobic activity. Some other markers that we were really interested in that emerged with a random forest asking which molecules best distinguish the different visits include diacetylspermidine, which is considered a marker of biofilms and is sometimes used in the context of colon cancer. And then also lactate, as I mentioned, is going down with visit 2. If you look at the metabolomic diversity with disease stage, you can see maybe a little bit more diversity in the earlier samples, and we have 3% of the variance attributed to disease stage. And the metabolomes also fall into 2 clusters. They're not the same as the clusters that form from the microbiome, which was maybe not my expectation. Going into this, I had this dream of like really nice concordance between the microbiome and the metabolome clusters. This might be a reflection that the host is donating a lot. A lot of the molecules we're talking about here are host derived, and figuring out whether they come from the host or the microbe is obviously at the edge of our knowledge at the moment. But we do see interesting differences in the diversity of the molecules between the clusters that form. And we have some indicators of those clusters, which are these molecules are starting to get familiar to you. Lactate and diacetylspermidine are also indicators of the metabolomic clusters. Okay, I can see I'm getting low on time here, which is, what happens when you try to show too many data slides. But here you can see we have some other molecules that are interesting indicators of disease stage on the metabolome side. And this is an unusual one that's more abundant earlier in disease stage, but we had a lot of molecules, especially amino acids, that are getting way more abundant with advanced disease stage. And we're not the only ones to see this. In fact, Deb Hogan has a really nice study showing increases in amino acid content in sputum correlated with worsening lung function. And so if you look at that across amino acids in our data set, it's a very consistent trend. So in conclusion, for our STOP2 work, we found subject-specific signatures explaining most of the variants for both the microbiome and the metabolome. But we do have metabolomic indicators of antibiotic treatment, efficacy, and disease stage emerging. So in theory, this could be used, for example, to validate whether antibiotic treatment is working. So where does this leave me now? What am I excited about these days? I had this I've started my lab 10 years ago, and I got to do the big study with tons of people with cystic fibrosis, which I really feel lucky that I got to do. But I have to say that my interest these days, a lot of them focus more on trying to engineer microbiomes. And so we have efforts in some of these areas using probiotics and prebiotics to shape microbiomes and using phages to target infections. So my group now has a lot of sewage samples, I have to say. We did some wastewater sequencing during the pandemic, and we have several projects working to collect and expand the host ranges of and evolve phages that target the various escape pathogens. Enterococcus and Stenotripomonas are our most studied ones in our lab. And we have fiber intervention studies. I've been doing this in my undergraduate classes, and also now I have a new cohort studying breast cancer survivors, and it's together with colleagues here at UCSF, actually. Laura Esserman and the I-SPY trial is our future hopeful goal, is to eventually help people who are recently diagnosed with cancer. There have been so many interesting correlations emerging that a high fiber diet associates with better cancer treatment outcomes, but we want to know if we can make that happen for everyone by introducing fiber, or is it something else that the people who eat a high fiber diet are already healthier and wealthier in some way? So our intervention right now is with chia seeds. That was actually not my idea. I did a survey of the students, and chia seeds were the most popular idea for the intervention, and they are amazing in terms of the fiber content. Look at this. Out of the 12 grams of carbs in a serving of chia seeds, almost 10 of them are fiber. Beans, it's about half. So it's a pretty amazing nutritional profile. And I've been working with our new teaching kitchen at UCI as well. And the audience of this seminar series we've developed, where I give a microbiome lecture and the chef makes a recipe that's relevant. And our audience is supposed to be the UC system, actually. It's supposed to be the staff of the UC system. And they're all recorded, but you're welcome to join. It's once a month, and it's a beautiful kitchen. It's like the Great British Baking Show. And she's an amazing chef. Every time I cook, I have like all kinds of memories of things she told me while I'm cooking. It's really fun. Okay, so thank you very much. I really appreciate your attention, and I'd welcome questions.
Thank you. All right. Well, thank you very much, Sumaya. It is an honor and a pleasure to be introduced by one of my superstar former trainees. If I could apply for a postdoc in her lab, I would, but I'm just not good enough at experiments to do it. So those of you out there who are looking for future training opportunities, I would work for Sumaya if she would take me, but she won't. The title of my talk is actually, we're going to talk about bacteria and phages in my talk. And we're going to explore Simone Biles. The title of my talk is Jump, Flip, Hide, Insights into Genomic Variation in the Microbiome. And as Sumaya mentioned, I think me and Tiffany have probably spent the most amount of time in this room of anybody in the audience. I calculated it, and I think between HSW 300, 301, 302, and 303, I spent over 2,400 hours in these classrooms. These are my disclosures. The most important slide, of course, is a big thanks to all of my scientific colleagues in the lab, past, present, and future. We've really had a wonderful time working together, exploring the wonderful world of microbiology. And I thought it would be useful to give you a little bit of a sense of who we are in the PUT lab. We are microbiologists. We like genomics. and we're interested in medicine. And so everything that we do functions at the intersection of these three fields. Those of you who follow our work or have had the chance to talk to me know that we love mobile genetic elements. Mobile genetic elements are drivers of microbial variation and evolution, and to date, they've been pretty difficult to study. And so over the course of the last 10 years, we followed technological advances and computational advances to try and understand how microbial genes jump, flip, and hide. As microbiome interested people and scientists in general, we like to measure stuff. I bet you measure stuff. I bet you think that what you measure matters. I do too. But if I'm honest with myself, I think I usually measure what's kind of convenient. I measure stuff that somebody else developed a ruler for. I measure things that other people have benchmarked. And as a consequence, we're often limited by our measurement tools. And so over the course of the past 10 years, we and many others in the field have tried to develop tools that push past those boundaries. We're really driven by the fundamental curiosity of what is in this picture. What are these? Capsules. These are fecal microbiota transplant capsules. This is actually a picture that I took. I'm really glad that I did. I've showed it lots of times. These are capsules that were developed by Libby Homan's group at Mass General Hospital. She's one of the people who actually drove the commercialization of these types of treatments. But of course, one of the challenges to these types of therapies, if I put on my physician hat for a moment, is that we don't really know what's inside them. These are complex mixtures of microbes, and really, it's just like looking out into the universe. Ideally, what we'd like to have is an orderly list of genomes that comes from these complex mixtures. And of course, one of the major tools, workhorse tools, that has enabled us to make a lot of progress in this space as a field is DNA sequencing. Of course, you can use short-read DNA sequencing, which has become increasingly cheap. It has advantages. It also has some disadvantages. And for those of us who are interested in mobile genetic elements, long-read sequencing has been quite transformational. And I'm going to explain a little bit about why that is the case. If you're interested in hearing a little bit about how we think about sequencing and how it can be used to explore new things in the microbiome, Yishai Pinto, a current postdoctoral fellow in my lab, who will be starting his own group at Bar-Ilan University later this year, wrote a really nice review on this topic. So how many of you have heard of MAGs, metagenome assembled genomes? The answer is everybody, because Emily talked about them this morning. But of course, you know, the idea is that we've got this complex mixture. We want to have an orderly list of genomes. It's like we go from the picture that I clip arted from the internet on the left to a Circos plot that gives you the genome of the organism on the right. That's what we want. For us, one of the things that has really vexed us is how good this is and how bad this is. And let's talk about that a little bit. So metagenome assembled genomes, or MAGs for short, have provided insight into uncultured organisms. And this has been a really important step forward in reference-free microbiome research. And we heard about this from Curtis earlier this morning in the context of things like viruses. We heard about it from Emily in the context of things that don't bin well. This is really work that I credit Gene Tyson and Jill Banfield with really kicking off in 2004. That was the first description of the idea of a metagenome assembled genome. And then really beautiful work from 3 different groups came out at the end of 2018 and early 2019 referenced here that really blew the field open. It just did it in force. So MAGs are really great because they can give us insight into microbes that we haven't yet cultured or we don't yet know how to. But the problem is, in reality, we end up with these incomplete and often highly fragmented genomes. So here's a Circos plot of a MAG that we generated in our lab many years ago. This is a Phascolactobacterium species. It's got 2 different colors of green and lots of breaks. So the breaks indicate the fragmentation, and the light green stuff didn't actually assemble into this bin. It didn't bin with the rest of the contigs. So this is an example, a pictorial, real example of a fragmented, incomplete genome. So, like, who cares? If it reads out as complete and not too contaminated, does it really matter? Well, I would argue that a lot of the fun is in this accessory genome, in the mobile genome. So let's take a look at a toy example. Imagine we have 2 contigs or 2 genomes. In this case, I've represented them as linear. We've got sequence A, sequence R, sequence D. These are contigs. Then we've got B, R, D. Now, in this case, R is shared between the two. So these are two different genomes that have the same sequence in them in a different context. If you were to do a short read assembly, what you'd end up getting spit out of your short read assembler of choice is A, B, C, D, and R all as separate contigs. But in reality, if you were to actually inspect the graph that underlied this, you would actually see this in the graph. So this is the toy graph. You would know that there's a repeated sequence R. 5' of it, you end up with A or B, and 3' of it, you have C or D, which means that there are 4 possible solutions to this toy example. You could have ARC, ARD, BRC, or BRD. Now, you may say, this is great, but who can give me an example of a repeated sequence in nature that happens in microbial genomes? This is going to be interactive.
CNG islands.
Okay, CNG islands. What else? And thank you, Sarkis. I will give you $20. Invertible promoters. What else? Ribosomal RNAs, actually, is one. You said ribozymes, but I'm going to extend that to ribosomal RNAs because it's convenient for me. Repeated sequences abound in nature. The 16S ribosomal RNA operon is often repeated multiple times in organisms. Might be related to the growth ability of an organism. There are gene duplications. These can be short-term gene duplications. They probably are happening in organisms that Peter is growing upstairs right now. There are insertion sequences or transposons, my super favorite type of repeatable element. And all of these are very, very common, and I would argue interesting and important. So along came long-read sequencing. And long-read sequencing has the ability to really bust through this problem because you can essentially get a sequence that goes right through the correct path. Now, this, of course, is a lot of people's work in many labs reduced into a simple slide. But this is really an example of a technological innovation that enabled us to do something that we couldn't do before. So Eli Moss, a former graduate student in the lab, and Dylan McGinney, also a former graduate student and former postdoc in the lab, teamed up to apply nanopore sequencing long ago when the technology was still fairly primitive, but good enough to get the work done. They applied nanopore sequencing, we did long-read assembly and polishing, and what we ended up with was something that I thought was super exciting. I remember when Eli showed this in our subgroup meeting, I stopped, I took a picture of the slide, and then I shed a single tear. I also shed a single tear when Stewart et al. Published the first linear p-copri genome from rumen using long-read sequencing, because this is really cool stuff. This is the genome. This is a prettied up picture. And what you can see on the outside track is just like a blue circle. And never in my life have I been so happy to see a blue circle. This was a complete closed genome. Inside, you can see 2 different tracks. Those are for the short read assembly and the read cloud assembly, and they're fragmented. And if you take a look on the very, very inside, you see a lot of tick marks. And these tick marks are very interesting because these represent the money. So these are insertion sequences. These are transposons, autonomous jumping genes. I prepared this slide around the Olympics last year, which was a happy time. And this is Tara Davis Woodhull, who got the gold medal for the long jump. So there are so many insertion sequences here. There are 127 insertion sequences in this organism of about 5 or 6 different flavors. So this is an example of why this genome actually evaded being closed because of these repetitive elements. So one of the things that we asked ourselves over the course of this time was we knew transposons were important, and we wanted to be able to use short read data because there's a lot more short read data out there, and we, like many of you, are data parasites, and we wanted to use these data to be able to locate MGEs. So long ago, in like 2019, 2018, Matt Durant, a former graduate student in the lab, developed a short read-based algorithm called MGE Finder that takes in short reads, single end or pair end as input, and the output is a list of integrated MGEs and their locations. We decided to apply this to this very, very cool experiment. Who here has seen the video on YouTube of Microbiome and Roy Kishoni's megaplate experiment? If you haven't seen it, do it right away, but not right now. It's really cool. They had like a huge, like table-sized agar plate. They plated E. coli on the outside, increasing concentrations of antibiotics on the little like vertical stripes that you can see. They took a video of the E. coli growing. As the E. coli grew, if they wanted to access more nutrients, they had to acquire antibiotic resistance. And that's actually what you're seeing as it starts to invade the center parts. They then took the video and they annotated where the resistant clones were arising, picked up some bacteria from there, sequenced them. They wrote a science paper about it. They got a lot of press. This video is actually in the Wellcome Trust Science Museum in London. So I recommend you go and you check it out. It's a very, very cool experiment. But what we did was something that basically just extended what they did a little bit. So what you see are different genes in the E. coli on the x-axis. You see the independent mutation events on the y-axis. So a lot of mutations, SNPs and indels were acquired as is represented in the gray. And that's what Michael and Roy reported. But of course, there were other things that happened as well that they missed because they didn't look for it. And we decided to look for it with MGE Finder. And one of the things that we found was not only were SNPs and indels arising, but there were insertion sequences, jumping genes that were moving around the genome. And that's what you see in the black bars. And so these are structural variations that are conferring antibiotic resistance in very short timescales. So you could ask yourself, when else is this happening? And we've done that several times in the lab. Most recently, Matt Grisham and Aaron Baird, two fantastic graduate students in the lab, have become very interested in comprehensively enumerating different types of insertion sequences, including those that are replicative. How many of you knew that there are copy-paste transposons? It's kind of amazing. So there are cut and paste transposons, there are copy-paste transposons, and now that we have long-read sequencing, we can enumerate them much more effectively. We can not only count them, but we can demonstrate where they are. Just to give you an example of what that looks like, this is like a very early unpolished experiment. If you were to use short read information alone and you were trying to annotate things like IS-256 and ISL3, if in Enterococcus faecium, you'd end up with a few things that are annotated. If you use MGE Finder, our short read approach on short read data, you see many more. But the most comprehensive measurement of these is actually with long reads. Now, there are some subtleties to this. We could talk about this slide for 2 hours, but instead we'll go right by it. And I'll say that there are a lot. And this is, a lot of insertion sequences. So what we decided to do was ask, is this a lot of, does E. faecium have an unusually high number of insertion sequences? Or is this just something that's present in all of these different bacteria and we've been missing them? So who here can tell me why we care about E. faecium? It's a pathogen, I think I heard. Yeah, so it's a pathogen. It's infecting people over in that hospital and in every hospital in the world. And it's interesting because it's evolving antibiotic resistance quite rapidly. It's become mostly vancomycin resistant. So this is just a process slide. All I want you to remember is we counted insertion sequences and where they were in all of the escape pathogens. These are important human pathogens. And what we found was these are the pathogens on the rows. E. faecium is a clear standout. So E. faecium has way more of these insertion sequences than other organisms. Why is this important? Well, one of the things that can happen when you have repetitive elements in a genome, if they're oriented in the same direction, they're called direct repeats, you can actually have deletion of sections of the genome. And this can lead to reductive genome evolution, where your genome becomes smaller and smaller. This has happened several times in the past. It has happened in some of these organisms that are present here. If you take a look at those that have transposase counts on the x-axis that are way outliers on the right, it has happened in Shigella. It's actually how E. coli evolved into Shigella. It's how Yersinia pestis evolved. And it's how Bordetella pertussis, the causative agent of whooping cough, evolved. So we think that Enterococcus faecium is going down this same path. And that just shows you all the outliers. So MGEs can disrupt genes. They can lead to reductive genome evolution, which can be important in pathogens. Sumaya, who you just met, wrote a really nice paper in 2020 with her colleagues Alex and Eli, where she demonstrated that these mobile genetic elements can move in clinical timescales, days, in patients in this hospital in their gut microbiomes and lead to adaptive evolution and antibiotic resistance. And of course, they can also do interesting things like recoding genes. So let's turn our attention to Simone Biles and invertons. We're going to talk about DNA flipping instead of people flipping. And this is work that was led by Patrick West, a former postdoctoral fellow in the lab, and Rachel Channon, a current postdoctoral fellow in the lab. This is a beautiful marriage of two minds in a way. Patrick is a computational biologist. Rachel is an outstanding experimental microbiologist. And I think this story really demonstrates how useful it is to collaborate across the aisle, as it were. So what is phase variation? Phase variation can describe actually two different things. It was described before we even knew about like DNA and genomes. It was described back in the old microbiology days. And nowadays we know that in bacteria, phase variation usually refers to either DNA flipping or to DNA methylation. For the purpose of today, because I work on genomic structural variation, we will not talk about methylation, although maybe in the future we will. So in this case, phase variation is when you have a region of DNA, as represented here in the light blue. It's bounded by inverted DNA repeats that are usually on the order of like 8 to 22 base pairs in red, and they can be flipped in place reversibly. As Sarkis mentioned, this is something that can happen in promoter elements, and it's known to flip promoters. And here's my favorite example of this. It's not actually the first example, but I really like it. This is in Bacteroides theta iota omicron, which is a model gut commensal that many of you are familiar with. B theta can choose which capsular polysaccharide coat it's going to wear. Sarkis's work long ago actually demonstrated that polysaccharide A on B fragilis was very immunogenic, and it's interesting in that context. But from a bacteriophage relationship perspective, these CPS coats can also determine whether or not a phage can infect the bacteria. So you can imagine the bacteria can benefit from flipping the sugar that's on the outside of its cell. In this particular case, this is a CPS1 locus. There are 8 different CPS choices in B theta. 5 of them are controlled by these promoters that are actually invertible, that are upstream of the locus. And so when this flips, now the promoter is oriented in the opposite direction of the CPS1 locus. We think that these invertases that are upstream of these are actually what are mediating the flipping. So what we asked was a simple question: can we identify new invertons in B theta in our clinical metagenomes? Putting on my medical hat, we collect thousands, we're literally probably now 10,000 plus samples of stool from patients undergoing hematopoietic cell transplantation. And we decided to apply a very clever tool that Xiaofang Zhang developed when she was in Eric Alm and Ron Xavier's group. It's a short read based tool. It's called Phase Finder. It takes in short read data and then it outputs invertons. Now, in her beautiful paper, she looked in intergenic regions for these flippable promoters. And all we did was a little tweak, which was to say, let's look everywhere, not just in intergenic regions. Let's look within genes, intragenically as well. With that one little tweak, in addition to finding over 350 invertons in B theta alone in these 400 clinical samples, we found 67 that were contained entirely within single genes. So these are within gene flips. So imagine what a flip could do. It could recode a gene. It could cause a premature stop codon. We found many examples of this that I'm going to blow through for time's sake, simply because this work is now published. But we found examples where you could actually have a flip that introduced a truncation in a gene like Thi-C. You actually get that truncated product transcribed and translated. We measured it at the RNA and protein level. There are also flips that don't cause a truncation. For example, in an extremely meta turn of events, there is an intragenic inverton inside a gene that flips DNA. So there's an intragenic inverton in CPS1 invertase. This is a validated missense intragenic inverton, so it just changes the sequence of the protein slightly. This is a multiple sequence alignment up at the top, and over below it, you can see two alpha-folded structures overlaid on one another. One is the forward direction, one is the reverse. The areas in the red and blue are the parts that are recoded. And you can see that you have a difference in the predicted structure. So we wanted to measure these, and I think I've already demonstrated to you that long reads are really well suited to identifying structural variations, much more so than short reads. And so we developed and applied a tool called FABA. The input are long reads, and the output is a list of these invertons. We then downloaded all of the 30,000 long read isolate genomes that were present, and we quantified a bunch of different versions of these invertons. And to make a long story short, we found lots of invertons across the prokaryotic tree of life. So in the last minute, I'm just going to say that in addition to being able to disrupt genes, upregulate genes, and recode genes, in recent work, we've done long-read sequencing of longitudinal stool metagenomic samples from several participants. This is work by Angela Hickey, who's sitting in the audience. And what we've been able to do are identify very interesting structural variations that aren't transposons, and they're not flips, they're actually integrated phages. So just to give you a sense of what we're able to do, this is an example of a short read assembly of a region of DNA. If you were to take long reads and do assembly of that same region, what you end up with is one big piece of assembled DNA. If you were then to use a tool like Vibrant or Genomad to call the phages, you can see that you get a very nice phage boundary. If you take a look at that phage boundary and you call genes within the predicted phage, you can actually identify an integrase. So we think that the integrase is a DNA recombination enzyme that's leading to the insertion of the DNA in this locus. You can look at longitudinal samples. These are 2 time points. So this is the same individual 2 years apart, metagenomic data. And what I hope you can appreciate is right at the phage boundary, you can see that at time point 1, about 75% of the bacteria has the phage inserted at that locus, whereas at time point 2, it's basically gone. This is really beautiful evidence over 2 years' time that the integrated boundaries are very clean. So now you can imagine that not only can we find definitively the host of this phage, but we can say, I think that that integrase targets this specific DNA site that we can recapitulate based on the sequencing data. And this is the type of work that we've been doing to essentially mine these double-strand DNA phage integrases, which use these enzymes to highly specifically and efficiently integrate into genomes. As you know, these phage genomes can be huge. We've actually found a jumbo phage in a manuscript that was published yesterday from our African microbiome work that's 220 kilobases that integrates using one of these types of enzymes. So it can integrate huge amounts of payload, and they can integrate into safe harbor sites. This is much more controlled than random integration, and you may ask yourself, this is starting to not sound like a microbiome talk. This is starting to sound like a genome engineering talk, and that indeed is one of the directions that we in the lab have taken and spun out into a company. So in work that was in collaboration with the Bintu Basic and Sue labs, we were able to actually mine these enzymes, demonstrated the work in mammalian cells, and are now trying to develop them for use for genome engineering. So you can do a lot with these long reads. I told you that microbiome data and microbiome research is still reference limited, and that long reads are a useful tool to study integrated MGEs. We like to enumerate and place insertion sequences, and we think that this will help us understand phenotypes of organisms and evolution of important pathogens. We talked about phase variation and how DNA inversions not only happen to promoters, but also within genes. And finally, we talked about long reads and how they can be used to predict prophage hosts, and we can mine the boundary information to develop new genome engineering tools. So as we move forward, we're going to develop and share large longitudinal data sets. We're interested in exploring the consequences of insertion sequence expansion. We'd like to understand how phase variation is regulated and deconvoluted specificity rules, and of course, map host range of phages and understand phage compatibility. So with that, I'd like to thank the lab, our many collaborators, our funders, our patients' families, and their care teams. And I'm happy to take a couple questions if we have time.
So our lab works on the effects of the microbiome on the immune system, I think as Ami referred to many years ago, but also more recently on the nervous system. And so we become quite interested in the gut-brain connection, if you will. And so this story that I'm about to tell started about 12 years ago when I came across some interesting literature, just anecdotally, that many Parkinson's patients have gastrointestinal symptoms. I'll go into it a little bit more. But I thought it was pretty curious that a classical neurological condition, neurodegenerative condition, had GI manifestations. And I thought to myself, of course, this was when like anything you did in the microbiome was, revolutionary. And we said, let's just try it. Let's see if the microbiome is a component of how the gut and the brain may communicate in Parkinson's disease. And so this is some of the evidence that I'm referring to is that, of course, PD is a movement disorder. It's characterized by rigidity, resting tremors, posture instability, deficits in gait, and other physical and motor symptoms. There are also many non-motor symptoms associated with Parkinson's, very, very well known, one of which is constipation. Up to 70% of individuals with PD will experience constipation many years before they're actually diagnosed with their movement disorder. And so here's just one schematic that constipation, REM behavioral sleep disorder, loss of smell, hyposmia are all prodromal symptoms that can be found, again, in a very high proportion of individuals with PD. And so, again, wondered whether or not the microbiome had some sort of a role in either the GI manifestations or, again, maybe even more intrepid in the brain manifestations of Parkinson's disease. And so, in fact, the literature goes back quite interestingly to 1817 when James Parkinson wrote an essay on shaking palsy. You guys can look this up. It's pretty interesting, where he talked about case studies of individuals that he was following. The disease was actually later named after him. He didn't name it himself. And he talked about people with essentially constipation and their shaking palsy, and treated them with diuretics and with essentially laxatives, and reported improvements in bowel symptoms and as well as movement symptoms. And so 20 years ago, or approximately 20 years ago, there's a hypothesis proposed that's called Brake's hypothesis by Heiko Brake, as a physiologist in Germany. And he was looking for the classical pathology of Parkinson's, which I'll describe in a second, and he found it almost everywhere, but in people who manifested very early, found a lot of these aggregates of this neural protein alpha-synuclein in the gastrointestinal tract. So that led to this hypothesis that maybe in some people, Parkinson's begins in the gut, or again, and other peripheral tissues as well. And so there's two leading hypotheses now, that it's either the translocation of this pathology from the enteric nervous system through the vagus nerve up into the brain and ultimately the brainstem that leads to the GI symptoms first, and then the motor symptoms next. And then again, once the symptoms or the pathology gets into the brain, it actually spreads throughout the brain. And this is actually a clinical staging parameters that are used in diagnosing people with PD, like to see how far the pathology has actually progressed. And so again, very much a hypothesis based on observation anecdotal data. Now we have just a lot of evidence in mice. I think it's pretty conclusive in animals that you can experimentally trigger Parkinson's disease by promoting pathology or even inflammation in the gut. You have to have the right genetic background in the animal to do so, and that pathology spreads in the enteric nervous system, and then most likely through the vagus nerve makes its way into the brainstem and into the brain. Epidemiologic data in people show that people who are vagotomized for like peptic ulcers or anxiety many years ago are statistically protected from Parkinson's. And so again, it's not we don't know for sure if Parkinson's ever begins in the gut. Again, a lot of compelling evidence that it does. And so there are now about 40 studies looking at the microbiome in Parkinson's. We published this paper a couple of years ago using two cohorts, one in Chicago, one throughout the United States, where we essentially just did shotgun metagenomics, as in collaboration with Rob, and showed that indeed there's a pretty stereotypic PD microbiome. You can see the PD individuals are here, household and population controls are here. And then what's really interesting to me though I haven't done a statistical analysis across all these papers, is that the same organisms keep coming up in different cohorts, whether you look in the US, in Japan, in China, and in Germany, where there's really extensive metagenomic data. You see, Enterobacteriaceae increasing, Akkermansia increasing, organisms like Faecalibacterium decreasing. So I highlight that here. I'm not going to show the data because it's a short talk, but what we've now shown is if we take Faecalibacterium prausnitzii, which you may know is depleted in inflammatory bowel disease, and we actually treat Parkinson's mice, we can improve their motor symptoms. So I think there's some immune modulation going on by organisms that are depleted in the Parkinson's microbiome. And so I alluded to the classical pathology of Parkinson's, aggregation of this neuronal protein called alpha-synuclein. And if autophagy and the proteasome or the lysosome don't clear these aggregates, they actually develop, and they essentially just clunk up the cell. They lead to cellular dysfunction, oxidative damage, and ultimately, over many years, into neurodegeneration and death of particular neurons. In Parkinson's, it's dopaminergic neurons that are the most vulnerable. The other neuronal populations are affected as well. And so this has been modeled in mice where there's a number of different mouse models of PD, but the one that we've used most extensively is an animal that overexpresses human alpha-synuclein. So it's known in people that promoter or enhancer mutations that just make more of the wild type alpha-synuclein lead to Parkinson's or predispose to Parkinson's. People who have chromosomal duplications, triplications in the locus that includes alpha-synuclein are also prone to Parkinson's. And so what this model is recapitulating is just elevating levels of wild type alpha-synuclein. And so you can see in this classical motor test, we just take a mouse and measure the time it takes to cross a beam. Mice that are overexpressing alpha-synuclein, so these alpha-synuclein overexpressing mice take longer to cross the beam than their wild type counterparts. And this is with an SPF or a laboratory microbiota. And so again, what we did in this paper many years ago is just deplete the microbiome of these animals. We rederive them germ-free, and you can see that their movement disorder is almost gone. It's across a number of different tests. On the pole descent test, you can see the same thing. In a test for fine motor skills, it's an adhesive removal where we put essentially a sticky on the nasal bridge of the animal and measure the time it takes to grasp and remove that adhesive, and you can see this deficit here. We run 5 different behavioral tests, and indeed in all of them, this mouse model shows a phenotype, and depleting the microbiome, even in mice that are aged, if you just deplete them with antibiotics, you can actually reverse their motor symptoms, which is pretty interesting. These mice also have GI symptoms, again, reminiscent of human Parkinson's, where the number of fecal pellets in a given amount of time, here in 15 minutes, is reduced in mice that have a complex microbiome, but not in germ-free animals. And in fact, the water content in these pellets is also reduced. So it's essentially like constipation. The pellets are drier. I'm not going to show it here, but gut retention time, bead expulsion through the rectum, gastric emptying are all affected in this animal model. So they really have very profound GI issues. And so, of course, classically, alpha-synuclein pathology has been studied in the brain. And so we want to look, do we see the same effects by depleting the microbiome? So here we're just staining brain sections that are affected. This is the cardia putamen. We see the same thing in the substantia nigra of these animals, where there's widespread alpha-synuclein aggregation. This is a conformation-specific antibody. You can see here in red. These are all aggregated forms of alpha-synuclein. The antibody doesn't recognize the monomer. And then in germ-free animals, you see almost no aggregation. There's no difference in gene expression. If we rederive the animals under germ-free conditions, there's no difference in the levels of the protein that's made. It's just that the protein aggregates less when there's no microbes. And again, it's the aggregation that leads to the pathology and to the disease. And so, we got a lot of mileage out of germ-free rederiving this animal model. We characterized, short-chain fatty acid levels and how, again, the pathology may spread to the brain. But then we also realized that this is a nice tool to test some of the correlations from human PD that, again, give you, very nice and compelling associations, but are not functional, right? And so what we did was we took microbiomes from individuals with Parkinson's and healthy controls and transplanted them into these germ-free mice that are prone to Parkinson's. So this is a review that Sue wrote many years ago. I use this slide often. It wasn't just for this symposium. I think this was with Olaf Peterson, where this really nice model, which I think it's aged well, suggests that there's something that changes the microbiome from a healthy to a dysbiotic state. It could be a lot of different things, right? And that disease in the context of a potentially a genetic predisposition in most cases then can manifest in disease. And then one way to test that is to then take the microbiome and transfer that into mice. And so here you're looking at the aggregate data of 6 pairs, healthy controls and Parkinson's. These are all household controls. So there's in every single case, there's a sex difference. But what you can see in the alpha-synuclein overexpressing animals is that mice that were colonized with a healthy control microbiome don't develop the worst motor symptoms as alpha-synuclein overexpressing mice that are colonized with the Parkinson's microbiome. The other thing that you'll see is that genotype does matter, at least in this context, that the wild type animals, regardless of which microbiome that you put in, don't develop these motor deficits. And so this suggests that indeed that human microbiome transplanted animals with alpha-synuclein overexpression leads to worse motor symptoms than microbiomes from healthy controls. So we started asking ourselves, so what is it about the Parkinson's microbiome that may be meeting this effect? And of course, the answer is probably many different things. And we've actually, I told you about the F. presnitzi story. We've worked on a couple of other organisms, in fact, molecules that are different between the PD and the healthy control microbiome. But we got interested in one aspect of what is unique about the PD microbiome, or at least different about the PD microbiome than the healthy controls, is that a number of papers have now shown that there's an increase in the load of bacterial amyloids. So alpha-synuclein, that neuronal protein that's involved in Parkinson's, is an amyloid-like protein. It's, again, I think many of you here obviously are familiar with the prion hypothesis that these proteins are self-aggregating. They induce, additional copies of that same protein to aggregate, and then those pathologies can spread, within a neuron, between neurons, and again, potentially even, between tissues. And so what's been known for many years is that bacteria have amyloids as well. And so levels of this bacterial amyloid CSGA are increased in the PD microbiome. So we didn't know this when we started this work, but now 5 different studies have shown that to be the case, that people with PD have higher levels of CSGA. So CSGA is a monomer of, or is a component of this filament called curli. So there's actually an EM of this E. coli cell, and these are the curli fibers, and they're involved in adhesion. A lot of these strains are pathogenic outside the guts. They're your pathogenic strains of E. coli. And what was interesting in our data set is, again, this is going back to the humanized mouse model, is that there's a higher load of CSGA in the mice that receive microbiomes from PD patients than that of healthy controls. So we thought to ourselves, maybe there's some interaction between this bacterial amyloid and this mammalian amyloid. And again, there's precedent for that as well, because we know that A beta and tau can interact with synuclein. So mammalian amyloids can cross-seed and affect aggregation of other mammalian amyloids. So we thought to ourselves, maybe a bacterial amyloid can substitute for mammalian amyloid and either initiate or accelerate aggregation of alpha-synuclein. And very recently, I was alluding to this, but studies in a Korean cohort, in a US cohort, a cohort in China, and another cohort that I previously mentioned in Japan, in Germany, have all shown the same thing, is that individuals with PD have higher levels of the genes that encode the operon for curli. Here's CSJ. This is the bacterial amyloid. And what's also interesting is that in this one particular study, in this Korean cohort, that they subclassified the PD patients as brain first or body first. And this is sort of a loose characterization. It's not a clinical diagnosis, but what it basically says is they go back in a patient's history. Were their neurological symptoms the first signs of PD, or were their gastrointestinal or peripheral symptoms the first signs? If the peripheral symptoms are the first signs, then that's body first. And you can see if you compare, at all of these components, there's more, gene copies in individuals that they characterize as body first, suggesting, again, it's correlation, but suggesting that maybe, something in the gut triggers alpha-synuclein aggregation, potentially of this bacterial amyloid, and that maybe that could be the initiating, or at least some of the first events involved in the disease trajectory that ultimately leads to a PD diagnosis. And so we showed in vitro, this is a biochemical reaction, just purified proteins, that CSGA, this bacterial amyloid, can accelerate synuclein aggregation. So this is the thioflavin T assay. You're looking at synuclein here. If we add a 25-to-1 ratio of synuclein to CSGA, you can see it's accelerated the aggregation of synuclein. It reduces the lag phase and the time to half max. And then ultimately the fibers themselves, whether they were formed just by synuclein or accelerated by CSGA. The fibers themselves look the same. And at this concentration, CSGA itself doesn't aggregate. So we're actually looking only at the aggregation of alpha-synuclein. And so we've done BioCore experiments. We can't really find a physical interaction. So others have shown that there may be transient interactions between CSGA and alpha-synuclein. So a number of different labs are now working on this biology. And so then we went back to the animal models. And so instead of putting in complex microbiomes here, we're just now monocolonizing, just for proof of concept, the same alpha-synuclein overexpressing mice with either wild type E. coli that make curli, that make CSGA, or curli mutant. And you can see, again, so the first two bars are essentially the data I've already shown you, or similar to data I've already shown you, is that these mice will develop motor symptoms if they're colonized with wild type E. coli. And really interesting, we've put like probably 10 different bugs into monocolonized into this exam models. None of them cause these symptoms other than E. coli that express curli. So again, there's probably other triggers in the microbiome that are quite interesting. But in this case, I think we can faithfully say that here the effects are curli mediated because the mutant E. coli, which colonizes the same levels, goes to the same place in the gut, doesn't cause the profound motor symptoms. And so this is the beam traversal, the pole descent, the adhesive removal test. This is Heinemann clasping. And so all of them, again, show a phenotype. We've also done experiments, it's in this 2020 paper, where we injected peptides of CSGA directly into the gut wall of these animals, a single injection by surgery. Those animals develop synuclein aggregates in the gut and synuclein aggregates in the brain and motor symptoms. So we do think that it's required and sufficient. Its molecule is required and sufficient. We've also injected mutants of CSGA that don't aggregate, that are amyloid deficient, and those recombinant proteins do not trigger this cascade of events. And so, as you may imagine, because these mice develop motor symptoms in the CSGA or curli-dependent manner, the synuclein pathology in the brain also forms. So there's more of these protease-resistant alpha-synuclein aggregates in the brains of mice that are colonized with the wild-type E. coli versus the curli mutant. And then if we look at this phosphorylated form of synuclein, which is a hallmark of pathological synuclein, you can see there's more of it in the substantia nigra. And in fact, in these regions that are positive for tyrosine hydroxylase, the enzyme that makes dopamine, which is really deficient in individuals with PD. And we also showed that the same strains can induce alpha-synuclein aggregates in the gut, at least if they contain the CSGA protein. So you're looking at neuronal tracts here in the colon, and everything in green is aggregated forms of alpha-synuclein, and you can see more of that in the soluble fraction, as well as there's more of the phosphorylated, the more pathogenic alpha-synuclein in the intestines of the animals exposed to this bacterial amyloid. And so overall, this suggests to us that, again, this bacterial amyloid may be part of the pathophysiology of PD in a subset of individuals. Looking across all the studies that have looked at the relationship between this bacterial amyloid and Parkinson's disease. So we roughly estimate that about 18 to 20% of people with PD that have dysbiosis, that have changes in their microbiomes, that elevation of CSGA or the curli operant is the defining feature of what makes the microbiome different than that of healthy control. So it's not going to be true in all PD patients, but again, maybe a fifth of individuals with PD, at least correlative, have higher levels of CSGA. And so we worked with Axial Therapeutics, a company I started many years ago, to develop specific inhibitors, drugs that prevent the aggregation of CSGA. So again, there's many, you may know, there's drugs, antibodies that are, and many companies that are developing therapies against aggregated forms of alpha-synuclein. But in a subset of individuals, again, if this is an initiating or an accelerating factor, maybe we can drug the gut instead of drugging the brain. Again, it's sort of a defining principle in the lab as well, is that if we identify bacterial targets, then it's much easier to get drugs into the gut than into the brain. And so AX5006 is an entirely novel molecule. It's completely synthesized by the company based on some principles that we defined through experiments. We found some natural products that were doing the same thing. So they took those chemical scaffolds and built on those and defined this entirely novel molecule. And through a series of molecules, and then tested which one was able to prevent synuclein aggregation. So this is, again, in a biochemical reaction. You can take CSGA, incubate it with alpha-synuclein, and then give them the drug. And then you can see in a dose-dependent manner, the alpha-synuclein aggregates don't form if you're inhibiting aggregation of CSGA. And the drug is quite interesting, the way the company developed the drug, is that they made the drug entirely gut restrictive. It doesn't cross the epithelium, it doesn't get into the circulation. It's only found the gut. So it essentially, as it's passing through the gastrointestinal tract, it finds its target and does its job. Because this is mouse studies, but they've done this in like pigs and dogs, that the drug itself does not enter into the circulation. Again, it's only found in the feces, which gives you added advantage of potentially less side effects once we're in the clinic. It also, the drug also prevents expression of CSGA. So it's well known, other people have studied that if CSGA itself doesn't aggregate on the surface of the bacteria, there's a feedback loop through a chaperone that then activates a small RNA that then suppresses transcription of CSGA. Bacteria don't make things that they don't need. And so here, just looking at CSGA mRNA in E. coli cells, you can see in a dose-dependent fashion, the drug actually prevents the production of CSGA through this same feedback mechanism. And the same in animals. So when we monocolonize the mice with the wild type E. coli, you can see that there's less CSGA expression in the mice that were given the drug versus the mice that were given controls. But of course, we wanted to understand, is this going to improve motor symptoms? And so we did a prophylactic study where we essentially started treating very, very early before the mice even developed motor symptoms, and we're able to prevent the development of motor symptoms. And we got a little bit more bold, if you will, and asked, can we reverse existing motor symptoms? So we aged mice to a point where we knew that they would develop motor and GI symptoms, and then started treating the animals. These mice were treated for 10 weeks, and then we retested them for motor symptoms. So this is pre-treatment. So you can see these animals have a beam traversal phenotype, and at 2 different concentrations, 50 and 150 mg per kg. You can see in the beam traversal test that these animals perform better after being on drug for 10 weeks. Same with the pole descent, same with the adhesive removal, which I think is quite interesting. And so again, I'll say, I'm not sure this is going to translate into the clinic, and I'll touch on that in a minute, but it's really, I think gratifying to see that we can reverse existing motor symptoms. And finally, the final data set is to look at actually alpha-synuclein aggregates in the brain. So again, this is sort of the holy grail, if you will, of neurodegeneration, at least of these amyloid-like proteins. Is once these aggregates form in the brain, is there any way to remove those plaques, or to remove that pathology and that damage? And so again, at the time point that we started treating these animals, they had alpha-synuclein aggregates. This is in the substantia nigra, and you can see after 10 weeks in a dose-dependent manner, we can actually reduce the burden of those pathologies. And so what I think is happening, at least in this animal model, is that we're still looking at a time point where the neurons are distressed, but they're not dying. In fact, we know that there isn't neurodegeneration at these early ages. But the mice and the neurons in these animals are under really, strong, dysfunction. And we believe dysfunction is coming from overactive mitochondria that are compensating for the pathology of alpha-synuclein. So as these aggregates are building up in the cell wants to compensate for its activity. So it actually boosts mitochondrial function. That mitochondrial function leads to reactive oxygen species that's causing the motor symptoms. And so we're getting reversal in the animal models, but again, I'm not sure we're going to see that in the clinic, but we'll wait to see. So this drug will actually go into people later this year. And so I'll conclude with our hypothesis that there is dysbiosis, and potentially a very characteristic dysbiosis in Parkinson's disease, and that potentially intervening at the level of the gut may be therapeutic. In people, we may hopefully slow or halt their progression. At least in mice, we can reverse their progression. And again, I showed you evidence about drugs. We've published on probiotics or high fiber diets in this model. And then we have an unpublished story on fecal transplants from healthy individuals and healthy mice into animals that already show symptoms. We can reverse their symptoms as well. And we think that that's inducing clearance mechanisms in the brain. And so I'll leave you with this concept that I alluded to a few minutes ago, is that potentially by understanding this gut-brain connection and understanding the etiology of diseases where the microbiome may be involved, that we can deliver drugs to the gut, much easier proposition than delivering drugs to the brain. I will end there and thank the people who did the work. Just the 2 people I'll shout out is Tim Sampson, who's now got his own lab at Emory. So he did all the Curli work. And then Matthias did all the work on the drug AX5006, our collaborators and our funding sources. Thank you.
Thank you so much for the kind intro and for the invitation here. I'm honored to be part of this wonderful and slightly intimidating lineup of speakers. So I first have to, of course, thank the people who are doing this work. So this is my lab at Northern Arizona University. Specifically, I'll talk about some work that Katie Kahn did. She's a PhD student in my lab. And if you knew me from my work here, or my time here, or the first few years of having my lab, again, my title might have been a little bit of a surprise for you. I got my start looking at host microbiome interactions at the airway mucosa, focusing on chronic rhinosinusitis, asthma, and cystic fibrosis. As our lab has grown, we've expanded our interest into the role of the microbiome in the gut microbiome in peripheral diseases, asthma, and neurodegenerative disease. So we know Alzheimer's disease is highly prevalent, 6th leading cause of death in the United States. Right now, more than 6 million people are living with Alzheimer's disease in the United States and 47 million people worldwide. As our aging population, for a little while here. As our aging population grows, we expect the rates of Alzheimer's disease to grow as well. The key disease pathologies are amyloid beta plaques and neurofibrillary tangles. So the plaques are extracellular deposits of amyloid beta protein, hallmark pathology, and then neurofibrillary tangles are intercellular aggregates of hyperphosphorylated tau protein, which disrupts neuronal function. Recently, though, the role of neuroinflammation has been important in recognized in Alzheimer's disease, and some key immune types that are dysregulated in AD are microglia. In health, they function to remove cellular debris. And in Alzheimer's disease, they typically tend to co-localize with A-beta plaques and become more activated. Astrocytes are involved in the maintenance of the blood-brain barrier. And in Alzheimer's disease, we see reactive astrocytosis or astrogliosis, which can result in impaired blood-brain barrier and decreased neurotransmitter uptake, and I'll talk about that in a little bit. The gut microbiota brain axis, as Sarkis had very beautifully described in Parkinson's disease, may well play a role in Alzheimer's disease too. So I'm just highlighting 2 studies that demonstrate an altered gut microbiome in Alzheimer's disease. The first study in the image that you're seeing on the screen is from a cohort of individuals with established cognitively impaired Alzheimer's disease and compared to matched healthy controls. And they did see a number of bacterial features that were altered in AD. Recently, another paper came out that demonstrated the gut microbiome composition is an important predictor in preclinical AD in individuals who are cognitively normal but display pathologies of Alzheimer's disease. They're a very well characterized cohort, I think, the Wisconsin ADRC. And in this study, they could use clinical parameters in a predictive model of AD and get relatively good predictions of individuals in a random forest classifier. But when they added gut microbiome features with the clinical genotype and CSF biomarker data, they vastly improved their machine learning predictive model. So I'll tell you a little bit about some of the work we've done. Some recent data that we're getting, and then where we're going from here. And I'm pretty excited about all of those, but especially where we're going. And so our first question was how the dynamic bacterial communities change over time in mice modeling AD pathologies compared to wild type mice. We, based on data that had already been published in severe or cognitively impaired Alzheimer's disease in some of these mouse models, we hypothesized that our gut microbiome compositions would associate with pathology develop and potentially contribute to neuroinflammation. So we published this a couple of years ago, and this is the work of Emily Borsum, who was a former PhD student in the lab and is now at the Allen Institute in Seattle. And so we used a model, the 3X transgenic model, which models A beta plaques at around 24 weeks, and then A beta plaques and tauopathy at around 52 weeks of age. And these mice have 3 transgenes of early onset Alzheimer's. So they're predicated to develop these pathologies at these time points. And so what Emily did is we wanted to know what the microbiome dynamics were, not just at these time points of pathology development, but what is going on in the microbiota as these mice go through these pathologies. And so we looked at a number of immune genes in the gut and in the brain. We focused on the hippocampus and frontal cortex of astrocyte reactivity, microgliosis, and some general markers of inflammation. And then we profiled the gut microbiome. So Emily collected fecal samples fortnightly for a year. So if you're a PhD student in my lab, that's what you get to do. And here's what we found. We found that relative gene expression of GFAP, which is a marker of astrogliosis, was upregulated in the colon at 24 weeks. So really at the start of the pathology development. And here I'll orient you, blue is our 3X mice and orange are wild type mice. And then at 52 weeks, even in the colon, we see increased gene expression of GFAP compared to our wild type mice at 52 weeks. In the hippocampus, we saw and we expected to see this increased GFAP, so reactive astroglia at 52 weeks in our 3X mice compared to the wild type mice. We saw general inflammation, IL-6 increased in the colon at 52 weeks in our 3X mice compared to the wild type mice. And then in the hippocampus, we saw upregulation of MERK1, which is a marker of microgliosis, as early as 24 weeks, which decreased but was still significant at 52 weeks in our 3X mice. Microbial features. So we were interested in the changes in the relative abundance across time between these 2 mouse strains. We saw Lactobacillus salivarius was increased early in the wild type mice, again here in orange. And so what you're seeing, these are volatility plots. The thick lines represent the mean within each strain, and the thin spaghetti lines represent individual mice. And you can see the trajectory of Lactobacillus salivarius over time across the 52-week experiment. Interesting, mostly absent in our 3X mice. Thericibacter, which is frequently differentially abundant in a number of neurodegenerative or neurological disease. We saw an increase prior to pathology development in our 3X mice, which was mostly low abundance in our wild type mice. Thericibacter is not the most well characterized. Katrine had a really nice paper looking at Thericibacter in the model as well. But again, it's pretty consistently observed in Alzheimer's patients and in mouse models of Alzheimer's pathology. And of interest, it's a consumer of serotonin. Sarkis had mentioned Akkermansia. We saw Akkermansia enriched in our 3X mice, modeling AD pathologies prior to pathology development. Again, this happens around 24 weeks, and then it kind of peters out in the gut microbiome. So if we didn't have this dense longitudinal sampling, we might have missed this Akkermansia signal, depending on when we sampled, say, at the end of our experiment. And then we also saw Bacteroides acidifaciens enriched in our 3X mice at the time as pathologies started to develop around 24 weeks. This is a member of the mouse gut microbiome, not necessarily found in the human gut, but it is closely related genetically to Bacteroides fragilis. And that'll be relevant for the second part of the talk. And so the final part of this paper is related to that study I mentioned where you could, or where including gut microbiome features improved prediction of individuals with preclinical AD prior to cognitive impairment. We found that at 8 weeks of age, we could accurately predict what strain the gut microbiome came from using a random forest machine learning classifier. We saw accuracy of 1.44 over baseline. So these findings support the findings in preclinical AD that pre-pathology microbiota, and what Sarkis had been talking about, that pre-pathology microbiota features are predictive of, in this case, mouse genotype. And so the conclusion of Emily's study, we found Akkermansia, we were interested in Akkermansia mucinophilia enrichment prior to pathology development. But we ended up focusing for the next part of our study on Bacteroides, because several species of Bacteroides, including Bacteroides fragilis, can putatively function as ecologically keystone species. Sarkis beautifully demonstrated several years ago neuromodulatory effects in a mouse model of characteristics of autism spectrum disorder. In AD, there's been a few studies showing that Bacteroides can actually activate microglia and induce Alzheimer's pathologies. Spoiler alert, we did not find this, but we're still interested in Bacteroides, partially because it's a potent producer of neurotransmitters, specifically the amino acid neurotransmitters GABA and glutamate. And there's increasing evidence that GABAergic signaling is dysregulated in AD. So GABA is a primary mammalian inhibitory neurotransmitter, and glutamate is its counterpart is the excitatory transmitter. And this imbalance in AD really interested me. And the fact that several bacteria, notably Bacteroides, Bifidobacterium, and Lactobacillus, are potent producers of GABA. Some studies have suggested that peripheral GABA interacts with the immune system and inhibits vagus nerve afferent signaling from the gut to the brain. So this figure is from a paper focused mostly on depression, but depression is a comorbid pathology with Alzheimer's disease. And so we're interested in whether GABA signaling influences any of the pathologies we see in our triple transgenic mouse model. And how does this happen? Is it signaling through the vagus nerve? Is it getting into the periphery and possibly crossing this impaired blood-brain barrier? And so my second graduate student victim, Katie, no, she's wonderful. She's been doing amazing work on this. We designed this study, another 52-week study, where we supplemented mice with 1 × 10⁹ colony forming Bacteroides fragilis suspended in sodium bicarb into applesauce. And then the vehicle control is 1 ml of the vehicle sodium bicarb in the applesauce. And what you do, and it's actually very cute, is you take your applesauce and you put a few of the food pellets on the floor or on the bottom of the cage, and you kind of spread the applesauce across the pellets, and the mice come in and scarf up their Bacteroides fragilis. And so that's the meaning of the apples in this. And then we had a number of outcomes, microbiome, targeted and untargeted metabolomics. We assessed neuroinflammation, but also GABA and glutamate receptor and transporters. QSIP will be the last part of my talk, and so I will explain it then. I have in italics, we assessed behavioral outcomes. The Morris-Water maze is a measure of spatial learning, and temporal object recognition is a measure of temporal memory. And then we measured GABA in the fecal and serum, and we're currently measuring it in the brain. The reason the others are in italics is because we saw no significant differences in any of our cognitive outcomes. But the caveat is, by the time we learned how to do these, bought a giant pool, got it set up in our animal facility, our mice we only had our 52-week mice left. And I think if we could have assessed this earlier, we might have seen interesting outcomes. Or at least that's what the optimist in me says. And so we found in our gene expression assays that Bacteroides fragilis, so I'll start with this first plot. And there's 2 other groups in this set of experiments. So orange are wild type genetic background mice given the vehicle control. Red are wild type mice given Bacteroides fragilis. Green are our 3X mice given the vehicle control. And then blue are our 3X mice given Bacteroides fragilis. And we see what we expect, increased astrocyte reactivity in our 3X mice given the vehicle control, but we do not see a significant increase in our 3X mice given Bacteroides fragilis. We also found, and this is all I should have mentioned in the first plot, this is all after correction for multiple comparisons. We looked at a transcription factor that regulates a lot of pathways associated with longevity, FOXO3. It's also inversely associated with glutamine synthase, which I sort of didn't describe this. See if I can go back. So GABA is synthesized by glutamate, by the gene glutaminate decarboxylase, and by the enzyme, and then glutamine and glutamate can be shunted back and forth by glutamate synthase. And so we saw a decrease in FOXO3 in our 3X mice treated with Bacteroides fragilis compared to the 3X mice treated with the vehicle control at 52 weeks of age. And then finally, we looked at this astrocyte glutamate uptake receptor, and we see a significant decrease in this glutamate uptake receptor, which is specifically expressed in astrocytes in our 3X mice given Bacteroides fragilis compared to the control at 52 weeks. We had also hypothesized that Bacteroides fragilis would modulate the gut microbiome composition. And this surprised me a little bit in that still the primary driver of separation is strain, but a secondary driver of separation is Bacteroides fragilis, but it wasn't, it didn't affect the gross microbial community as much as I might have hypothesized. So we see the greatest separation at 8 weeks of age. Again, this is prior to the development of AD pathologies or neuroinflammation. And at 24 weeks of age, we start to see these communities converge a little bit, but our 3X mice in blue given Bacteroides fragilis shift a little bit towards the wild type mice. When we look at the microbial features that are changing over time, again, Katie bravely accepted the task of collecting fortnightly fecal samples for the entire year of this experiment, even though I told her we've already kind of done this, you don't have to. But she wanted to, and I agree with her because what was interesting is we see early colonization of Bacteroides fragilis in our 3X mice here in blue. And remember, in our original sort of observational study, we didn't see Bacteroides fragilis emerge until about 24 or 25 weeks. And then later, around 24 or 25 weeks, we see colonization of Bacteroides fragilis at a lower level in our wild type mice. But interesting, this species was notably absent in any of our mice given the vehicle control. So it's not a part of at least our cohort, our lot or breeders gut microbiota. Bacteroides species that were classified down to species level, but unclassified at the species, were enriched in our wild type mice, regardless of being colonized with Bacteroides fragilis, and these were absent in our 3X mice in both conditions. And I did mention GABA. So we are looking at concentrations of GABA and using ELISA and our measuring GABA in the serum. It's a little inconsistent. The only trend that really popped out that was somewhat interesting was that we see the spike in our 24-week wild type mice given Bacteroides fragilis. And that sort of corresponds with this when it actually starts to colonize the gut microbiome. Our ELISA, we're optimizing these, but also we're working with our wonderful collaborator, Allegra Aaron at Denver University now. We're doing untargeted metabolomics, but she's also optimizing an assay that will target GABA and glutamate, and it's coming along, but we're looking at concentrations of GABA and glutamine in the fecal microbiome, or in the fecal sample serum and the brain samples that we have sent her. And on our untargeted metabolomics, we keep seeing this pattern emerge, which I find interesting in a lot of metabolomic features that we're looking at individually. These are fecal taurine conjugated bile acids, and they're elevated in our 3X mice given vehicle control here and here at 8 weeks of age. And this phenotype is rescued when we treat mice with Bacteroides fragilis, even though they are triple transgenic mice. And so taurine is thought to play a neuroprotective role in the brain, and previous studies have shown that increased taurine in the serum of healthy individuals. We're thinking maybe Bacteroides contributes to bile acid deconjugation of these tauroconjugated bile acids through the bile salt hydrolase. But this is just a hypothesis at this point. But importantly, this also mirrors when changes in the microbiome are most predominant, suggesting that maybe these microbe-derived or microbe-driven host metabolites are critical to the development of pathologies or protection. So the bacteria that I'm talking about are relatively rare in abundance in the microbiota. And we wanted to know really how can we gain insights into the actual contributions of these potentially rare taxa, which could be missed in standard amplicon or metagenomic sequencing approaches. And so Emily had mentioned differing tools in environmental microbiome research and human microbiome research. And I wanted to use some of your tools. And so what we're doing is using quantitative stable isotope probing in a mammalian model. This technique was developed at NAU, the Center for Ecosystem Science and Society, and it uses 18O label or isotopically enriched water as a universal substrate to quantify microbial growth and death rates. So some of the limitations of current amplicon or shotgun metagenomic sequencing, although we've seen, can provide extremely important and novel insights, but they're compositional. And all abundances are relative, so the population dynamics can be kind of unclear. And so what you can do is label DNA. The 18O DNA will get incorporated, or the 18O water will get incorporated into the DNA of microbes that are actively dividing and replicating. This hadn't been done in a mammalian model. We have 2 studies in which we're incorporating it. I didn't show you this in our study design, but in our Bacteroides intervention, we actually did supplement 6 to 7 mice within each group with 18O water, so we could look at microbial community dynamics using quantitative stable isotope probing. And so we've shown for the first time that we actually can label gut microbes using this technique. So this is a density gradient. The heavier curve indicates more incorporation of 18O into that DNA. And we can label the fecal microbiome, the cecum. We've struggled with other body parts using our current protocol. And so what this can tell us is we can look at microbial features that incorporate 18O, so that would shift to the right in this plot, more 18O atom fraction excess. And then the final point I'll make is, because I got my warning, what we can see here in this kind of excites me is that we would expect maybe that the more abundant microbial community members would take up more of the 18O. But what we're actually seeing, it's the rare microbial communities and members in the gut that are incorporating more 18O. So rare microbial communities or microbial features are plotted here, and then 18O atom excess is on the y-axis. So we're actually able to pick up the activities of rare microbial communities. And so, brief summary, we're really interested in pursuing more pursuing more studies with Bacteroides fragilis and gaining a deeper understanding in the mechanistic pathways by which it might contribute to Alzheimer's disease. And I have to thank all of our wonderful collaborators.
Yeah, and thank you for the invitation to do this. When you and Susan and Peter sent the invitation to speak here, as you recall, I immediately said yes, and it's wonderful to be here at the Benioff Center with such wonderful friends and collaborators. But also, I congratulate you on the Feast of Science that you put together today. With such a wonderful collection of speakers, many of whom are also longstanding friends and collaborators. So I've really enjoyed the day's science. Thank you. So I guess we're going to conclude this feast with a bit of a smorgasbord of a bunch of technology, but also in some historical context. And so what I'm going to talk about is some of the work we've done over the years to scale microbiome studies to address global problems. And so this is going to be a brief tour over the last 20 years of microbiome research. I have some disclosures, none are particularly relevant to this talk, except I used AI for a bunch of the pictures. Microbes are intimately involved in many of Earth's problems, as we heard from Emily earlier today on the environmental side, and as we've heard from many people in terms of infectious disease, chronic disease like Parkinson's and Alzheimer's, food production, pollution, climate change. And harnessing microbes to address these problems really requires better data and better quantitative approaches. And that's really what we've been focused on for the last 2 decades. It's not just the planet that's suffering, it's our own internal microbiomes as well. And so Marty Blaser, another close friend and colleague, documents this wonderfully in his book, Missing Microbes, where he highlights how as we brought infectious disease under control during the 20th century, at the same time there's been this explosion of chronic diseases, where when this graph was published back in 2002, no one knew any of those diseases were even connected to the gut microbiome in any way, whereas today we know that all 4 of those and dozens of others are associated with the microbiome in humans, and that you can cause or cure analogs of them in animal models by manipulating the microbiome, as we've seen very elegantly, for example, in Sarkis's talk and in Emily's talk. And then there's the problems that we didn't even know we had. So I want to highlight this paper from Nature Micro 2 years ago that we wrote a commentary on that basically shows that if you use standard host depletion, you can reconstruct your subject's genome from the human DNA reads that are left behind in there. And this isn't just a problem for people studying the human microbiome. In the same month, this paper in Nature, Ecology and Evolution showed that if you think you're doing environmental sampling, most of those environmental samples are contaminated with human DNA as well, that you can use for identification if you decide to do that. So think about all these poor ecologists who've never done human subjects research, never taken the training, don't even know what the letters IRB stand for, and now suddenly they've got all this potentially identifying human data. So just at the end of last week, our paper resolving this problem came out, where we essentially worked with the authors of the authors of the Japanese paper on the human problem, and showed that we can resolve this issue by better host filtering. But not only does it protect your patient's privacy, it also allows you to get the correct results. So previous versions of the human genome were highly incomplete in the Y chromosome. And for example, that can lead to erroneous conclusions about differences in samples. Here showing you what looks like a split between females and males in the cancer microbiome. Whereas if you do the host filtering correctly, all of those biases disappear. And I'm happy to say that Sergio was a co-author on this paper and helped refine it tremendously from a clinical perspective. So there's all these problems. Some of them are old, some of them are new. We need better microbiome solutions to them. And microbiome research is scaling dramatically from what's been a data-limited field to what I'd argue is now limited by the analysis that we can do. And I'm going to talk about what I see as 3 major transitions in microbiome research, 2 that have already happened and 1 that we're going through now. And the first of these transitions was integrating evolution and community ecology. I'm going to go through this pretty quickly because I know it's familiar to a lot of you, but it might be interesting perspective for the students. So 25 years ago, when I was in grad school, microbiome analysis looked like this, where essentially we got tens of bytes of data per sample, where each lane on this gel is a sample. And we use these techniques to separate out versions of the 16S ribosomal RNA using an alphabet soup of techniques that have now been forgotten. Like, you won't learn how to do these in the lab. You won't be able to publish them anywhere if you do them. But essentially, we could tell that there was differences in diversity there, right? You can see some have more or fewer bands. You can see there's differences in evenness. And even at that level, we could start to do community ecology, largely stealing from the landscape ecologists like Whitaker's work in the 1960s, where the concepts that you see on many of the posters, like alpha diversity, the diversity of organisms within one patch in that landscape, the beta diversity, or the dissimilarity between two of those patches, and even the gamma diversity, the diversity of the whole landscape, that's used less in microbial ecology. But we did do it once when we bought this blender to homogenize whole rats, so we could get the gamma diversity of an entire organism. These concepts were really dominated research for a decade, where we could look at all these diversity characteristics. And a key advance was to combine it with Norm Pace's concept that we should be putting all of this in a phylogenetic context rather than just counting species. Look at where those species were in the phylogenetic tree. And this is what led to the workflow that you're all very familiar with, where you get samples and extract the DNA from them, amplify, sequence, and then match them up to what's similar in GenBank. Ironically, this got less and less useful as there was more and more stuff in GenBank, because instead of hitting E. coli or B. subtilis or something else you recognized, you'd hit some unidentifiable long string of letters and numbers from a culture-independent study. And so we had to start building alignments and phylogenies to be able to slot in the new sequences. And so Kathy Lozapone, one of my first grad students at Boulder, exploited this idea to compare communities in this metric called UniFrac, which we've seen a few times today, where essentially we exploit evolution to compare communities. And so what we do is we take that phylogenetic tree, we measure pairwise the dissimilarities between communities on that phylogenetic tree, and then we can use multivariate techniques like principal coordinates analysis and clustering. And the very first thing we used this for was a paper that was led by Ruth Ley, but that Peter Tenbaugh was also a very valuable co-author on, where what we were able to do was basically look at a model of obesity in mice. And what we saw using UniFrac was immediately this very strong effect of family, which is why we now use household controls in what you saw in Sarkis' study, and also what we're doing in IMS-MS with Sergio, where those cage effects are important in mice, but they turn out to be very important in humans as well. And complementary to that, looking at the relative abundance, we were able to see differences in the microbiome systematically between the lean and the obese mice. But microbiome research in this area was really heroic, right? Like the 5,000 sequences collected in the study cost millions of dollars and took years to put together. And so I think about these heroic efforts as kind of being like the Lewis and Clark expedition, which took 45 people to cross 8,000 miles, and it took them over 2 years to do it, and some of them died. And you think about this and you imagine, what if Sacagawea could have used GPS, right? It would have been much happier for all of them. And again, tracing that expedition that took years back then, if you do it on Google Maps now, what you'll see is that today it just takes you 3 days to drive there, and it costs you $750 instead of 1.2 million, at least with current gas prices. But if you think a little bit about the infrastructure that supports all this, it requires GPS satellites, which are fairly expensive. It requires interstates, which are a shocking $30 million a mile to build, apparently. I had no idea about this. But then if you think about the benefits, like not dying, right, you could argue that that's priceless. And so a lot of what motivates us is building the infrastructure to turn these kinds of things from heroic efforts into things that are routine and just something that you do, rather than something that you have to prepare for years to be able to do. And our first real foray into this was in 2008, where we're able to double overnight the total number of 16S sequences that have been collected by the scientific community. And so what we did was basically introduce the now standard technique of coding digital barcodes onto the primers for amplifying 16S. In this first study, we looked at the lung of cystic fibrosis patients. We looked at rivers, we looked at the air, we even looked at the most complex microbiome we know of on Earth, the Guerrero Negro microbial mat. And when we put them together and cluster them with Unifrac, you can see the sample type clustering is beautiful. I won't say anything about the details of what we saw in each habitat type, because what I want to focus on is the cost. And so the 500,000 sequences that we collected in the study, the Sanger facility down the hall from my lab in Boulder was charging $8 a sequence at the time. So if we did it that way, it would have cost $4 million. Whereas we did it overnight on the then new 454 technology for $12,000. And this played a key role in enabling the Human Microbiome Project, the Earth Microbiome Project, many other large-scale projects. But what's important to keep in mind is that amount of sequencing that cost $4 million in 2007, cost $12,000 in 2008. Today, if I walk across campus to IGM and do it there on the NovaSeq, costs just 75 cents, right? And so the ability to reduce the cost of sequencing data has been amazing. Now, before you think you can sequence your stuff for 75 cents a sample, the problem now is that all the other parts of doing the project are more expensive than the sequencing itself. So a lot of what we're doing at the moment, and I'll come to this at the end, is reducing the costs of those other parts of the project. And so this enabled things like the Earth Microbiome Project, which I think you're probably all familiar with, where it was initiated by the DOE, but then ultimately paid for by private sources. And in this one project, basically from the meta-analysis that Kathy did right after she did that study on the lean and obese mice I showed you, all of the samples in GenBank that we could get that were environmental samples where we could tell what sample the 16S was linked to covered 202 samples and 21,000 sequences. Whereas just 10 years later in the EMP, we did 27,000 samples with 2.2 billion sequences, which is just an amazing expansion of capability in a decade, especially when you consider this as one study rather than a meta-analysis of everything in the literature. And we had to put together a huge number of tools to do this, including many of the standards that allowed us to do it. I want to say a bit about the computational improvements that enabled this. So the initial version of Unifrac, we thought 5,000 sequences split across a couple of dozen samples was a lot, and it was for its time. Then Micah Hamady in 2009 introduced what we heuristically called fast Unifrac that allowed us to process hundreds of samples. And then we optimized that further in 2015. Parallel fast Unifrac that we used for the EMP got us up to 27,000 samples. Striped Unifrac, introduced by Daniel McDonald, got us up to over 100,000. And most recently, we've been collaborating with Igor Svigoli at the San Diego Supercomputer Center to scale it up to over 1 million. And to give you a sense of this is just showing you a bunch of different implementations of Unifrac. So these will all give you the exact same result, right? But the algorithms for how you achieve that result are different. And this is on a log scale, so it's a little bit difficult to interpret. But basically what you're seeing is a couple of orders of magnitude difference out at 10,000 samples. But these algorithms scale badly. So when you get up to 1.2 million samples, what you can see is the widening of that gap between the newest implementations like GPU Unifrac and the older methods. And like I said, this is a log scale. So let me just put this in perspective. What we're talking about is if you did it the original way, it would take you 3,100 years to run this analysis, which is a bit longer than the length of your average NIH grant back when they were giving out grants, right? Whereas the improvement with GPU Unifrac is that it takes a day, which is a lot more feasible if you want to run something like this. And if you want to see what 1.2 million samples looks like, we can manipulate them in a web browser with the latest version of Emperor, so you can brush the metadata in real time, rotate it, all the stuff that you're used to, and the Emperor interface within QIIME 2. So we found out a lot of cool stuff in the EMP that I'm not going to tell you about, although I will highlight the importance of the ontologies that we developed there. And this is why additional efforts like what NMDC are promoting are especially valuable for getting all of the stuff into a structured format. And one thing we did here is we asked the microbes directly what they cared about and used clustering to develop those ontology terms rather than trying to do it based on first principles, which have largely led to a lot of unpleasant surprises. We also, 2 years ago, added multiomics to this, redoing a bunch of the samples with shotgun metagenomics and with metabolomics. And we're very interested in working to scale this up as the metagenomics and metabolomics protocols have advanced tremendously in just the last few years. So the potential to really get a sense of what this crosstalk across the planet is and all of these microbes and the molecules that they produce is possible to address like it never has been before. So anyway, all this community ecology stuff is great, but there's a few drawbacks when you have especially ecologists interacting with clinicians, where the clinicians tend to have a lot of questions like, you know, what do all those dots on those principal coordinate spots actually mean? You know, what are the axes, that kind of thing. What are all these different kinds of diversity? Like, can't you just have one measure for diversity? Why do I have to learn about Shannon and Simpson and a dozen other things? Which organisms are really making the difference here, which is especially frustrating if you're doing something on a PKA plot at the community level. And then the real killer, is this a clinical test, which everything I'm showing you here is research, and I'll say a bit more about that in the context of COVID. But basically, these ecological approaches are great, and they can show that the microbiome is associated with the disease by distinguishing case and control groups. Again, as you've seen in a lot of posters here today and in a lot of the talks, but they can't answer the categories of questions that you have to be able to answer when you're working with clinicians, or for environmental settings, if you're working in a regulated space with policymakers, which includes even things like looking at pollution from fecal coliform on the beach. So the second transition was to integrate machine learning, and this happened about 12 years ago now, and we can now build predictive models for a lot of different traits related to health from the microbiome. And so from that 2005 paper, you know that I've been working on obesity for a long time. And we can tell with high confidence, like 90% accuracy, if you're lean or obese based solely on the DNA of the microbes in your stool. So on the one hand, this is pretty cool from a technical perspective. On the other hand, from a clinical or a commercial perspective, it's not very useful because you can tell which of these people is obese without sequencing anything at all, right? But on the other hand, if you try to do that, lean or obese classification task from human genes, you can only do it with 57% accuracy versus the 90% accuracy we get from microbial genes. As Dan Knights showed as part of his thesis back in 2011, he's now a full professor at the University of Minnesota, doing a lot of really exciting work. But this general strategy of basically taking cases and controls and then building predictive models with random forests or SVMs or artificial neural nets or other techniques like that has been enormously successful in asking, is the microbiome linked to a particular disease? And can I fish out the specific microbial taxa that are involved in this linkage? And again, you've seen a lot of this today with things like AUC plots and other displays linked to this kind of classification technique. And so we now know that the microbiome affects a lot of processes through your body, with positive effects ranging from digestion to the brain, again, as we've seen here today, and with negative effects, again, ranging from the gut all the way to the brain. And so a lot of this work has been based on this machine learning paradigm, which has been very successful. So one really groundbreaking advance was this work in 2015 from Aran Segal and Aran Alnab's groups at the Weizmann Institute, where they used an elaboration of this machine learning approach to predict why do different people respond differently to the same food items. And so what they did is they basically took 800 people, hooked them up to continuous glucose monitoring, fed them a defined sequence of diets, and then read out what is the glycemic index personally for each food item, for each individual, during the study. And then to validate it, they took another 100 people, used their predictive model to build a good or a bad diet for those people, and then showed that they could either send their blood glucose haywire or keep it under control, depending on that designed diet. And what they found is that when they averaged the glycemic index values, they were very consistent with published values. But when they looked at them individually, they were all over the map. And so for most people, you'd think of rice as being a pretty healthy meal. What they shockingly found, and this is in their graphical abstract, for a fairly sizable fraction of their population, it was worse for their blood sugar to eat rice than it was to eat ice cream. And out of all the factors they measured, the microbes were really the deciding factor. And so we now know that the microbiome affects the personalized impact of a lot of drugs, especially Peter Tenbau's really elegant work on digoxin metabolism, followed by a whole lot of other drugs, has really established this. And so some of the examples range from painkillers to cardiac drugs to anti-cancer drugs, and especially checkpoint inhibitors, whether you're a responder or a non-responder, depends tremendously on the microbes that you have in your gut. And so what we're fascinated by is this concept that most molecules that we ingest, whether they're drugs or food, may have a tremendous role both in affecting the microbiome themselves and also where your response is affected by what microbes you have. So this is something, although I love DNA sequencing, this is something that I thought we couldn't get at with DNA sequencing alone. And one of the big motivations to move to UCSD a little over 10 years ago was to work very closely with Peter Dorrestein to really, if you will, marry the metagenomics techniques we've been working to mass spec. And I think we can agree that this has all been quite beautiful. And we started this on March 10, 2015 with our first joint lab meeting, where every single week since then, we basically got his lab and my lab together in a room to build up a cohort of people who care about both these things. I'm not sure if she's still here, but I saw Alison Vibernak, one of our grad students here earlier today, and seeing people like that go on to great careers has been a really beautiful part of this program. But for a while, we've been turning to citizen science to understand microbiomes, and I'm sure you're all familiar with the American Gut Project, which has been largely paused during COVID for reasons I'll mention briefly if I have time. But is now back up and running as the MicroZeta initiative. The idea is that microbes are acting as the Rosetta Stone to understand hard problems in health and the environment. And so we complemented this with this global foodomics project to run thousands of food items through the mass spec and see what was really in them. And what I'm showing you here is molecular networking data. So each dot on this is a molecule. We draw an edge if they're similar by a cosine score above 0.7. Then we color them according to source. And you can see a whole lot of green on this plot, which is stuff that was in food, and a whole lot of brown, which is stuff that came out the other end, obviously. And there's not very much blue. So what that is molecules that were found both in food and in feces. And this is just showing you how complete that biotransformation is as your food goes through your GI tract. And so again, we teamed up with Peter's lab, and this is work that Alison contributed to as well, where we could image microbes and molecules throughout the whole body of a mouse. Actually, Sarkis was also a key collaborator on this, providing germ-free mice that we could compare with SPF. So we basically scanned them in a small animal MRI, diced them up into little pieces, and then for each voxel, we ran them through sequencing in the mass spec and reconstructed them so that you could see the spatial distribution. And as one example, soya saponin is something that comes in on the soy diet. And so you can see it accumulating in Sarkis's germ-free mice at the top there in red, but it doesn't accumulate in the SPF mice to anything like the same degree. Whereas microbes encode an enzyme that clips off soya sapogenol, which you can see in the SPF mice, but not in the germ-free mice because the mouse genome does not encode that enzyme. So we thought that was pretty cool, but our reviewers at Nature disagreed, and they made us rewrite the entire paper about the bile acids, where we discovered several new bile acids that had different spatial distributions depending on whether or not you had microbes. And that's ultimately what the paper was about as published. In fact, and I'll share I'll share this with you because you're friends. The reviewers made us cut out all of the spatial data and all of the spatial figures And Peter snuck them back in at the page-proof stage as having been inadvertently omitted during the production process. So that's why all the cool pictures about what the paper was actually about are in there. But this led to just the beginning of a huge expansion of what we know about new bile acids and how different things get attached to them by your microbes, and the role that has. So we've been extending this in the area of Alzheimer's specifically, and I won't say too much about this because you just had a great presentation from Emily about the microbiome and Alzheimer's. But essentially here, we're piggybacking on dietary trials in humans to look at diets that are known to modify Alzheimer's risk and Alzheimer's impact. And then we're looking at the microbiome and metabolome consequences. There's also an animal model component of this led by Sarkis and our other co-conspirators, Rima Kadara-Dauk at Duke University, with a whole lot of other people and institutions involved. So the one specific aspect of this I want to highlight, because it's something you can really use in your own research, is this paper we published in Nature Biotech out of the project on this new concept called reference data-driven metabolomics, which uses the foodomics data as our primary initial example, but we're extending it to drugs and other aspects of the exposome now. And so the issue is that if you run a sample through the mass spec of blood or of stool, typically 95% of the molecules you see are unidentified. So what we did is we took this reference database from all those foods. We can say, well, I still don't know this molecule, what this molecule is, but I know it only comes from apples or from salmon or from onions or whatever. So now I can match up all the counts that are specific to specific food items and then use that as something that I can associate with the microbiome or associate with phenotype. So ground truthing this, you can see these are not microbes, but communities of food counts that you're seeing basically on a PCA. You can see it beautifully separates the omnivores from the vegetarians. And then the changes in the direction you'd expect, where the omnivores are eating more dairy and meat, the vegetarians are eating more beans and stuff. So then what we can do is we can correlate that. And here I'm showing you the correlations with 4 subscores of the standard memory test. But you can do this with any quantitative phenotype that you measure, right? And so what we're going to do is we're just going to rank the foods from best to worst in terms of the overall forgetting on this memory test. And so the foods that are worst are sugarcane and soda. The ones that are best are fish, strawberry, and mushrooms in this particular analysis. And you might say, well, so what? You could have picked any foods out of a hat and it would have been just as convincing. But when we looked at the literature, what we saw was this giant literature on how bad soda is for your brain. Also a giant literature on how bad sugar is from your brain and its specific links to Alzheimer's. And then at the other end of the scale, there's a huge literature on how good fish is for your brain, including specifically in the context of Alzheimer's. And then also we found some literature both for strawberries and for mushrooms, again, linking them specifically to benefits in Alzheimer's. So what's cool about this is that we essentially took thousands of person years of research and all those individual heroic Lewis and Clark style papers looking at one molecule or one category of molecules and turned it into something we could do in an afternoon on the mass spec, the equivalent of that GPS satellite. And the ability to take this for all kinds of other conditions and look at what foods are really contributing to them, we think is really going to crack open a lot of these problems with personalized nutrition. So this is the sort of thing you can do with machine learning, and it's great. But again, interacting with clinicians has not gone so well on this stuff. So they tend to have questions like, what does classifier accuracy mean? And do I really have to memorize all these acronyms? I tell them you should be good at that because you went through med school and you had to memorize all this stuff, but that doesn't fly. They ask things like, are these features causal, where not only are they typically not causal, but you have a lot of masking, especially in random forests, where you get one out of correlated set of features coming up as the one that it picks. Again, you get questions like which species are really making the difference, which is problematic if your input data doesn't even map onto species concepts very well. And then the real kicker, how do I explain this to patients? Which, if they don't understand it, getting it in a form where they can explain it to patients is even more of a challenge. And the base problem is that machine learning is hard and it can't answer key questions that you have about all this stuff. So that's what we were chugging along doing up until 2020. And like many other people, our lives were upended by the pandemic. And at the Center for Microbiome Innovation, we had a whole lot of cool technology for reading out DNA. We very rapidly partnered with others across campus and across the Mesa to retool that whole infrastructure to look at RNA instead, because SARS-CoV-2 has an RNA genome, not a DNA genome. And so we founded the Excite Lab, which is basically a new CLIA lab that does clinical testing initially just for SARS-CoV-2, but now we've expanded so we're certified to be able to offer any high complexity molecular testing related to microbes. And so essentially what we did is we took the lab pipeline, which I'm sure you're very familiar with if you've toured a similar facility, and adapted all the protocols for RNA. In April of 2020, we were already doing our first pilot run with National University as a drive-through site, and Rady Children's, who were already certified to run the NP assay. And we also expanded it into wastewater. So Smriti Karthikeyan, who was an incredibly talented postdoc in my lab, and who has now gone on to a faculty position at Caltech, which was just wonderful to see, realized that we could also do things at an unprecedented scale on our campus if we use better technology. So we set up a system with over, at its largest point, it got to over 150 active autosamplers on campus, looking at SARS-CoV-2 at the individual building level, rather than at the wastewater treatment plant or the city level, which is what it had been looked at before. And essentially what made this possible was a new mag-bead protocol, where instead of having to do peg precipitation in multiple liters of material, which typically takes overnight to a couple of days, we turned this into a mag-bead protocol we could do in a couple of hours. So for samples that arrived in the lab at 9 a.m., we were able to provide results for hundreds of samples by 3 p.m. the same day. And so we set this up as a campus map where you could just look at your building and see, was someone in there, was there a COVID signal in the wastewater yesterday? We continued to evolve that dashboard, so now you can look at all the historical data, although the program has largely been shut down due to lack of funding at this point. But we were able to show that this led to early detection of 85% of the cases on campus at one point. And we couple this to a vending machine system where anyone could just get a qPCR test, have that processed and Excite the same day. The results would go into your EPIC record, and we could cross-reference all of the clinical data with all of the wastewater data, which led to Smriti's 2022 paper in Nature. And one thing that was really cool about this is that we saw earlier appearances of variants of concern in the wastewater than we saw in the clinical samples for a whole succession of waves of variants of the virus as they came through. We also set all the stuff up at the county level with our local wastewater treatment plants and a public dashboard where you can basically see overall spikes and also the displacement of one viral variant after another. And this is still running. So even to this day, they're not collecting the caseload data anymore, but you can still see the viral variants as they're processed every week. So anyway, this was a rather large disruption for our centre that we're now in the process of recovering from and getting back to what we used to do. But it was really valuable for exposing what are the gaps that we need to do if we're going to do a clinical test for real, rather than doing research in the lab. Because the SARS-CoV-2 assay, we had that up and running in the lab within 2 weeks, but then it took us almost a year to get clear approval for it, and then to run over 1 million iterations of that test. So thinking about what we can do for real in the microbiome space is really motivating at the moment, and coming out of this experience, as well as a lot of the technology we were exposed to. So this leads to the third transition, which I'd argue is just starting to happen in microbiome science now, which is integrating AI. And I know you're sick of everyone saying that they use AI now, but we've been doing it for a while. So we launched the Artificial Intelligence for Healthy Living Centre that I co-directed at UCSD back in 2017 as a partnership with IBM as part of the AI Horizons Network. But I will admit it's only really picked up in the last few years. And I'm going to go from the population level down to just one guy for a moment, if you'll indulge me in that. So I'm not violating HIPAA because Larry Samara is the least private individual that there is on the planet. And he's been telling the story to everyone, including the Atlantic, and releasing all the data on the internet. And he's been releasing his own data on the internet for the last 25 years, starting with one variable of his weight and then adding dozens of variables with blood tests. So, for example, here's a whole bunch of his stuff on his display over at Cal IT2 that he calls Future Patient. Dozens of dimensions with blood tests, thousands of dimensions with Fitbits and calorie counters and all that kind of thing, and ultimately millions of dimensions with the microbiome. And so shortly after I got to UCSD, I was explaining the HMP data, which was then relatively new to Larry, on the huge display wall over at Cal ITT. And he said, well, you know, Rob, I'm a very interesting microbiome character myself because I have IBD actually. And what can I do to get myself on that map? So shortly after that, this box showed up in my lab, the first one of many. And he had actually been working for a while on getting his microbiome sequenced at that point. And so his data from 2012 to 2015 looked like this, if you just plot the taxonomy. And don't worry if you don't see anything in there, because neither did Larry, despite spending some ungodly amount of compute and personnel effort to get that plot. But when we reprocessed it with CHIME and Unifrac, this is just rotating the data frame, but you can see that there's these clear clusters. When I animate it through time, you can see that he starts off at the edge of the blue region and travels through the blue, crosses over to the red, and then bounces around at random in the red. And so again, you might say, so what? But we can link that to all his clinical data. And what we see is that the initial shift was caused by a course of antibiotics. And then in this period where he's losing weight and his health is getting worse, this blue balls period, as he calls it, you can see that he's traveling directionally in the PKA plot. And then he changes his meds, which correlates with that transition there. And then when he's apparently bouncing around at random in the red, his weight goes back up to a healthy set point as his IBD symptoms resolve. And the idea is that if we have these personal trajectories for a whole lot of people, we'll be able to give you much more precise answers about where your microbiome is going and what you should do with it than if we just have the occasional snapshot. So we've collected a whole lot of additional samples from Larry, and this is just going up to 2019. And even at the taxonomy plot, some things like he wound up having to have a resection surgery, which you can see by that arrow actually has a big enough impact that you can see it even in taxonomy. But Larry was an avid follower of the American Gut Project, and one of the key results in that is that we saw that a lot of different factors affect the microbiome, but plant diversity, just as simple as the number of species of plants that you eat each week, has a much larger effect size than many things that we thought would be more important, like, for example, how old you are, whether you took antibiotics recently, and even whether you have IBD. And collaborating with Peter, we also showed that this is true for the metabolome. So if you eat more kinds of plants, you'll have a more diverse metabolome as well. Anyway, you might have seen this on Hack Your Health recently if you watched that documentary. Larry decided to increase his microbiome diversity by making this green smoothie and consuming 60 plant species every single day. And if you're interested in the actual ingredients list, we did put it up on the MicroZeta website, but I don't recommend it. I tasted it once; that was enough. So I was really inspired by Katie's story about collaborating with chefs, which we urgently need to do for this project. And yeah, so like I mentioned, this was on Hack Your Health recently. If you want to see it on Netflix instead of for me. But you know, one of the big challenges is interpreting the data, which, as you can see, completely baffled the host. Again, the key to understanding this is to relate it to the clinical data. So basically, he was experiencing this period of weight gain and transitioning into metabolic syndrome. And the last 3 months of this red period, he had gone vegan and gluten-free for 3 months, and it had no effect on that trajectory. He switched over to drinking the green smoothie and then also, unfortunately, time-restricted eating, which is confounded with it, which is the sort of thing you have to deal with in N=1 studies. But what you can see is that that change immediately got his blood glucose back under control and kept it there for years afterwards. And this was coupled to a 42% drop in his total fat, and also coupled to a 36% drop in his visceral fat specifically. So large phenotypic effects, although unfortunately just a sample size of one person. And what's really remarkable about this is if you've been following the microbiome literature, you'll know that most diet interventions, you basically keep looking like yourself after the diet intervention. It's very difficult to change your diet so much that you look like a different person. But Larry actually managed to do it. And over this time series, he's become 5 different people, 2 of them before the surgery, 1 after the surgery, and then with a vegan gluten-free period and with a time-restricted feeding period. So what's cool about this is that with extreme enough interventions, you can get differences that are larger than the standing variation between different individuals. So what we're doing is we're building this into a digital twin model. And if you're not familiar with this, it's a concept that's very widely used in engineering and manufacturing. So for example, many cars today essentially use predictive models based on the sensor networks throughout the car to warn you about things that are going to happen to your car based on that predictive model and get you to take it to the shop before it fails rather than afterwards. And for example, that was deployed across the whole Tesla fleet. In terms of applying it to living cells, the first attempt to do this was back in 2012, looking at a multi-scale network within one cell. And then more recently, we've been collaborating with Enos Telia's group in Galway with Larry's data to essentially look at the stool samples and use the Agora 2 pipeline to simultaneously model the host metabolism and the microbial metabolism together. And so this is starting, so being able to feed in different dietary parameters and so on into that model is just starting to yield results. We're doing it at the moment. With about 50 metagenomic samples, and we are about to do it with the whole time series. But the problem is that most of the data that I showed you was either 16S or short-read shotgun metagenomics. And the quality of the digital twin that we can make of Larry right now is unfortunately not very good, right? It's going to be pretty pixelated and low res. Whereas what we want to be able to do is something that's, beautiful, high res, upscaled, contextualizes him in his environment. And so to do this, we've been collaborating with my former grad student, Jake Minnick, who's now a postdoc with Todd Michaels at Salk, where we've been switching over to PacBio sequencing from short-read Illumina. And as Ami told you already with the data she showed on Oxford Nanopore, the long-read sequencing is transformative. So we get dozens of complete genomes assembled out of every single sample that we look at, rather than the fragments that you're used to if you've been doing mags from short-read. And so we're currently in the process of switching all of our pipelines over either to this method or to synthetic long-read methods like we're using with Sergio on the latest iteration of IMS-MS, which have the advantage that you can get much deeper coverage for the same cost, which is important if you want to look at rare taxa. So then the question is, if sequencing is about to get dramatically and ridiculously better, what do I do with all my legacy data? And so John Kirkland in my lab has been looking at transfer learning for upscaling using an attention model, which is related to the kinds of models that LLMs use. And essentially what we're using is ASVs as an input variable into an encoder block. And then we map feature embeddings into unifrac space, and then we relate that across ASVs and metagenomes where we have paired samples. And so the transfer learning that we get between the predicted and the actual GOTUs turns out to be very good. And here what you're looking at is a superposition between actual shotgun metagenomic data in yellow and the predicted values for each of those samples from the 16S data on the same samples, using Green Genes 2 as an interchange layer, as many of you have seen me present on before. And so the really cool thing is you can see a whole lot of pairs with one red and one yellow dot, which is showing you the exact same sample on 16S upscaled as what you would have got if you had read out the metagenome directly. So we're really excited about this as a way to rescue all the legacy data that people have collected to date. And we're also using this technology in a large-scale project called NPH, or Nutrition for Precision Health, which is an ancillary project of all of us. And the goal of this project is essentially to develop algorithms to predict individual responses to foods and dietary patterns. And this is a fairly large-scale investment from NIH, kind of like the HVP that Curtis talked about. The study design consists of 3 modules. So the first module is taking 10,000 people cross-sectionally, measuring their microbiomes, and then asking, does their microbiome at baseline predict their response to a nutritional challenge? Module 2 and Module 3 are longitudinal analyses. One's prescribed diets at home, and the other is diets in controlled settings in clinical centers. And then there's a whole lot of information being collected about them, including quantitative analyses of both the DNA and the RNA of every stool sample, as well as both targeted and non-targeted metabolomics. And this data set is already becoming available on the All of Us workbench to anyone if you want to get involved in analyzing it. We're also using AI now for a bunch of other stuff. I'm not sure if this demo is actually going to work, but let me see if yeah. So what we're looking at here is basically the challenge of cleaning up metadata. And so this is a Jupyter notebook.
Time initiate rest.
That you can just talk to and get it to clean up the metadata.
Do any of these values look like they refer to the same time period? Write code to replace within 1 hour of birth with less fun. Now run it. Write code to show unique values of time initiate rest. Now run it.
So basically the potential to do this kind of thing where you interact with no coding skills in natural language with a notebook to clean up your metadata, we think is going to be tremendously valuable for reducing the barrier to entry on a lot of these microbiome studies. So producing AI-ready data at scale also requires a lot of improvements on the wet lab side, and I want to make sure there's time for questions, so I'll just abbreviate this. But as many of you know, the way we do microbiome studies at the moment is a lot like loading up a ship in 1950, where everything's a weird shape, it's all custom, you have to fit it together, requires a whole lot of dock workers, multiple days to do it. Whereas what we want to do is we want to make it like a container ship, which is basically why you can get all that cheap stuff from China and so forth. And it's cheap even when you ship it across the ocean. And so what we're doing is we're transitioning from the heterogeneous tube formats that we've had to deal with in the past to this new system based on the matrix tubes that we first interacted with when we were setting up the COVID lab. And so together with Peter, and this was led by Katrina Brennan, a very talented grad student in my lab, we now have a single tube extraction method where we can basically take racks of those matrix tubes. We do the extraction for both DNA and metabolite single tube, which dramatically reduces cross-well contamination. And then you suck off the supernatant and put it in a separate plate. And so you can do DNA and metabolite analysis of the same samples at scale. And just to reinforce how cool it is to do this in high throughput, for example, for scanning the tubes to accession them, if you have a spreadsheet where the barcodes that are pre-labeled on the tubes are matched up to your metadata, maybe cleaned up in that notebook I showed you, you can just scan it, and then all of that connects to our Cheetah database, pulls down the metadata, and you can start looking at where your stuff is arrayed on the plate. Thank you. Even things like decapping are tremendously facilitated by automation. So that's a 96-well rack of tubes. And those of you who do lab stuff are probably imagining unscrewing the first tube, which you're done with about now, and going on to the second tube and starting to unscrew that one, and going on to the third tube and starting to unscrew that one, and so on. Whereas what you can see is that the instrument has already done all 96 in the time that I could maybe do 4 by hand, and it's done them perfectly. And so the idea that you can just feed rack after rack of tubes into this and get your microbiome and metabolome data produced at scale is what we're working towards very rapidly. And we've already deployed this throughout the MicroZeta initiative. So if you're interested in doing projects using this infrastructure, definitely send me an email. And we have more and more projects, including clinical ones, using this infrastructure. So one way to put a lot of the stuff that I've told you about today succinctly is that a large part of the challenge now for microbiome studies is not so much doing the lab work, but it's building a user interface for our microbiomes that allows us to interact with them in a usable way. And so our dream with this is that we can do it in a way that is useful for ordinary people. So maybe as you're faced with 1,000 yogurts in the supermarket, the idea is that you might be able to get something in real time from your microbiome data that would help you make a choice about that. And so we've started to develop this already. I'm sure all of you have used ChatGPT by this point. We're calling this something related but slightly different, where the idea is that you should be able to ask your phone in natural language, things like, should I eat this product to improve my health? Or maybe things like, can you highlight with AR the products that I should eat instead of this? And if you think that's ridiculous, I challenge you to take your phone and use Google Lens to take a little photo of an orange or an apple or something, because it will recognize that as an apple and tell you all the nutritional information on it immediately. And so a lot of the technology to do this stuff exists already. It's just a matter of linking up the right databases. And so our dream is to do this sort of thing, but make it not work just here, but throughout the world. So preterm birth is a huge issue for the Gates Foundation, for example. And the idea is that perhaps you would be able to do the same sort of thing at a produce stand in India or Bangladesh. And if that sounds ridiculous, smartphone penetration, not cell phone penetration, which is essentially complete, but smartphone penetration in India is projected to be 95% by 2040. And Bangladesh is only a few percentage points behind. So if we develop this technology now, it's not going to be that long before the entire world can use it, just as so many other technological advances related to mobile have already been adopted globally. So anyway, that's the kind of crazy stuff we're up to at the Center for Microbiome Innovation with a bunch of company partners, a bunch of faculty. We'd certainly encourage you to join us and join us for that CIMM meeting in March that Sue was generous enough to advertise here. But what we're really excited about is the possibility that AI may finally deliver what physicians want in a way that machine learning and community ecology have failed. We think that the translation and upscaling of different kinds of microbiome data are going to get around this issue of different methods being used to produce irreconcilable results in different studies. We think that explainable and accurate and personalized predictions are going to be really revolutionary for how you interface with the microbiome data. I didn't talk about this, but we're very excited about the possibility of generative AI to design new microbiomes and diets and therapies. And then the natural language interaction that I showed you a very early version of in that Jupyter notebook, but which we're deploying throughout our workflow. We think that's going to make all of these techniques not only accessible to a small priesthood, but really to democratize them so anyone interested in adding a microbiome component to their study will be able to do it. And so I'm confident that these advances are going to transform our ability to use our microbiomes. Now, just in the last 1 minute, I want to pull it back to global scale. And I talked a lot about food. I'm still surprised by how many people haven't seen this figure, which was published in 2019, which essentially relates how bad a particular food is for you on the x-axis in terms of its relative risk of mortality versus how bad it is on the y-axis in terms of its average environmental impact. And that's a straight line correlation there. That's pretty good. If I give you more resolution on what the actual data points are, it's relatively unsurprising. But the really exciting part to me is that environmental problems are a huge issue. However, especially in the United States in the next 4 years, I don't see anything being done about them. But given this correlation, what if we could trick people into doing things that were good for the planet because they were good for their own individual health? And I think optimizing these systems jointly to optimize planetary health as well as human health offers a lot of potential. So with that, I have a huge number of people to thank in my lab, in Peter's lab, and the CMI, and a huge number of collaborators, many of whom I mentioned, but I couldn't mention you all. So I apologize if you're here and I left you out. And then finally, thanks again for the invitation to be here. I'd be delighted to answer any questions you have, or if you're interested in any new collaborations using any of this technology stack or other stuff I didn't talk about here, we're very actively looking for new collaborations and ways to work together with other centers, including the Benioff Center, to jointly approach third parties for new sources of funding that do not rely on the federal government. So certainly love to talk more about that over dinner. Anyway, thanks again. Looking forward to any questions you have.